There we go. Okay, now we're recording. So I am stuck in Houston, Texas now because of that missed flight. Instead of enjoying the 70-degree weather, I am enjoying 105-degree weather. I'm not bitter. I'm a little bitter, but still, we are based in Houston, and we are excited to be there with you guys. The Taylor Group is a bench-to-bedside, full-service phage therapy group. We go out and we collect samples when we get a patient isolate, and we look for phage that can kill the bacteria. We analyze and make sure we analyze the phage and make sure this phage is safe to use in a person, and then we formulate it on site to give to the medical doctors treating the patient, and it gets to the patient that way. We've had great success, and if you've had a chance, I'm not sure when Austin, Dr. Twilliger's talk is, but he gave a talk on our success so far over the last few years. Now, the objectives of this workshop, I was going to joke around and say that we were going to talk about the mathematical basis and theoretical basis for bioinformatics, but this joke works much better if I'm there in person, when I can stop you from leaving. And I even practiced my Randy Savage voice, but sadly, that fire ruined all of this. We're not going to do that. We're not focusing on the mathematics or the theory behind bioinformatics. What we're focused on is understanding intuitively how these basic bioinformatic analyses work. This is kind of a special topic for me because I was actually trained as a bench scientist. I did my PhD work as a molecular biologist and geneticist. And I found that as I was going through that, I was both better and I enjoyed analytics and bioinformatics more. And so I switched gears halfway or towards the end of my graduate work and started focusing more on analytics. And since then, I've been kind of retrained as a bioinformatician. So this workshop is kind of what I wish I could have told myself when I first got started. You know, through grad school, I took certain courses that went over some of these concepts. I was familiar with the names maybe, but I didn't really understand how most of them worked, and I couldn't tweak the parameters or find new programs to use based on what I knew previously. So why is bioinformatics important? I don't think I really have to tell anyone here that DNA, RNA, proteins are the language of life. I think we all understand that, and understanding this language is essential for understanding life. And this is genetics. This isn't bioinformatics, right? This is just genetics. But since the genomic revolution, we have had to deal with a ton of data, and this has brought a lot of new challenges. As of 2019, there are over 200 million sequences in NCBI. And dealing with this amount of data and relating this data, parts of data to other parts of data, this is informatics, and specifically bioinformatics, the study of information, biological information. Now, you have your gene of interest. Now what? If you're a bench scientist, you could do everything yourself. You could take your gene, you could figure out what it does if you're working with phage, what it does with phage, how it may affect phage's ability to affect a host or other hosts, and how it changes how the host acts. If you're working with higher organisms, you can look at how it affects, cellular and organ networks. With whatever you're working with, you can then study the evolution, you can solve the structure, you can do all of this yourself. And, after 80, 90 years, when maybe you start to scratch the surface, you might have a full, complete understanding of what you're working with. What bioinformatics allows us to do is to take what we know or what we don't know and relate it to what we do know. And that's essentially what we're going to be doing with sequence analysis and bioinformatics. Is now you have your gene of interest. You don't necessarily have to go and do all these experiments if you can use scientific methods to relate your protein to another protein. Then you can just go and verify the function or hypothesize what the function is being or what the changes can mean between your protein and proteins that have already been studied. So in essence, it's almost a lazy way to get to where you want to go. It's a shortcut. The problem is bioinformatics can be very daunting, especially if you're not used to it or if you haven't specifically been trained in it. I mean, even looking at these different graphs and these different figures, I mean, they're crazy. I didn't know what half this stuff was when I first started getting into analytics. And it can be very daunting to look at that and feel like you're expected to know what that means and what that does. But luckily, like most things, once you start to understand some of the basics, things start to come together, and the higher order information, the higher order analyses build a lot upon the basic ones. So once you understand the basic analyses, you can start to understand and work out how the higher stuff works. So bioinformatics really encompasses a lot of different types of analyses we can do. I mean, everything from epigenetics, transcriptomics, molecular modeling. This all falls under bioinformatics. But what we're going to be focusing on today is really the basics of sequence analysis. Sequence analysis is really the analysis of DNA, RNA, and protein sequences. Now, the reason we're focusing on sequence analysis, because in bioinformatics, the term sequence analysis talks about identifying patterns so that we can use that to identify important elements, like maybe disease associations or parts that we know are involved in protein-protein interactions, conserved domains, functional domains. And like I said before, this is how you can take your gene of interest and relate it to what's already known, what's already out there, what's already been experimented on. But why are we focusing on the sequence? Well, if you look at the hierarchy of RNA, DNA, and protein, there is, or if you look at the structures, there's a hierarchy here. You have your primary structure, which is essentially the sequence. This is your straight sequence. For DNA, it's your ATCG. For amino acids, it's your peptides. And from there, you can build, the secondary structure is built upon that primary structure, how the primary structures interact with themselves from the secondary structure. This is the base pairing in DNA. This is stem loops in RNA, and this is beta sheets, now helices in amino acids. And that you can build on to get the tertiary structure. For proteins, this is the full protein structure. For DNA, this is the different types of helices, the classic B form that most people know, along with more exotic forms like the A form and the Z form. And then finally, the quaternary structure, which is how these different molecules interact with other molecules. For proteins, it's how protein subunits interact with other protein subunits, or with DNA or other molecules. And for DNA, it's how DNA interacts with the other pieces of DNA, or how DNA interact with protein. But it all comes back down to the primary structure, the sequence analysis. It's all built upon the sequence. The sequence determines the secondary structure determines the tertiary structure, and the tertiary structure determines the quaternary structure. Now, there's several types of sequence analysis, and you can do these on both DNA and protein and some RNA. We're not going to talk as much about RNA here. We're going to focus mostly on DNA and protein because a lot of the things you do with DNA translate well to RNA. But as complicated as this looks, really, it's all sequence alignment. A better way to show this table would be to show something like this. This is how all these different analyses are built upon sequence alignment. If you're going to do a variant analysis, you've got to be able to align your sequences. If you're going to do a functional prediction, you've got to be able to take the sequence you have, relate it to sequences that are known, and relate the function that way. So this is where we're going to be focusing. So bioinformatics, under the umbrella of bioinformatics, we're focusing on sequence analysis. And under sequence analysis, we're focusing on sequence alignment. Now, luckily, within sequence alignment, most programs can be broken down into just a handful of well-known and well-used algorithms. Some of the first and the ones that are most well-known are called dynamic programming. And we're going to go over the global pairwise alignments, which seeks to align the entirety of two sequences. We're going to talk about local pairwise alignment, which aligns regions of high similarity between two sequences and not necessarily the whole sequence. We're going to look at heuristic type alignments like BLAST and BLAST-like algorithms. And then we're going to look at how we can use this to build multiple sequence alignments, because, you know, pairwise alignment, we're aligning two sequences. With BLAST, we're aligning two sequences. But we can build upon that to build multiple sequence alignments. And we're going to talk about the two main types of those, progressive and iterative. And then finally, we're going to end with hidden Markov models. And as we go through this, we are going to look at programs and do some hands-on stuff with programs. So if you have a protein sequence or a DNA sequence, feel free to follow along and add that. And in the website, I'm going to have QR codes you can scan if you're on your phone. I also have PubMed IDs if you want to look at some of the papers I'm going to show. And I'll have the links that you can follow. Okay. Now, the reason we want to focus on these types of alignment algorithms is that most software use some combination of these algorithms. If you understand how these work, you're going to have an intuitive sense of how most alignment programs work. And we'll see that later on, how we can look at new programs and see how they use BLAST P, or they use dynamic global programming, something like that. Now, before we get into that kind of the meat and bones of these algorithms, we're going to introduce a dot matrix or a dot plot. And I'm going to introduce this because we're going to be talking a lot about matrices. And this is a really good introduction and a really good way to look at sequence alignments in like a two-dimensional field. Basically, the dot matrix is not an algorithm. You can use algorithms to make them, but it's a visualization method. And I find it's really helpful to go over this because it helps you visualize how you can align two sequences in a matrix. So when we have our matrix, which is just a table, we have our coordinates. And I'm always going to label these I and J, I for the columns and J for the rows. And this is because this is kind of convention in bioinformatics. Generally, the algorithms will use I and J to denote the rows and their columns. You can just think of this simply as if you're working with Excel, it works the same way as your ABC columns and your 1, 2, 3 rows. But also, or I think I got it backwards, 1, 2, 3 rows or columns and ABC rows. But I don't know if this is a dated reference for some people at this point, but you'd also think of it as kind of like a battleship grid. If you ever played that game as a kid, it works the same way. However you want to think of these grids, it works the same way. That's how you find your coordinates. That's how you find cells. Now, if you want to do a dot matrix by hand, you can first start by setting up your sequences across your columns and across your rows. Here, we're just going to do a very simple 5 nucleotide sequence alignment to demonstrate how this is done. And it's very simple, starting with the second row, because the first rows are going to be empty and the first column is going to be empty. You simply add a dot where you have cells that have matching nucleotides, or if you're using proteins, matching proteins. So in this case, when we go to the first one, we go across and we look for an A. When we get an A, we add a dot for the first row. Then we move down a row. We have a T. Where we see T's, we add dots. And again, on the third row, we have a T, so we add a dot where we have T's. C, we have no C's in the alignment or in the other sequence we're trying to align to, so no dots on this row. Then finally, we have an A again. Now, the important part that makes this really something you can use to build alignment is that now you connect dots diagonally from upper left to bottom right. And when you do this, you can see the places that alignments could be. I know this is very simple and very kind of elementary, but I wanted to start here in case anyone's not familiar with thinking about it in these types of terms. But this is how you can build an alignment by hand. There's additional statistics you can do with this, but we're not going to focus on that because dot matrices and dot plots are used kind of in more specialty terms now. They're used a lot for like larger alignments when you can't necessarily visualize all the information. You can get a good dot plot between like a genome to genome alignment and see where parts are missing and where they're similar between two divergent genomes. But now that we've started to introduce you to matrix, now we can start talking about how these alignment algorithms work behind the scenes, starting with dynamic programming. Now, there are two types of alignments, as I mentioned before. There is global alignments, and this attempts to align the entirety of a sequence from the first base or the first residue to the last. And then there are local alignments. These attempt to align sequences in just pieces or blocks of high similarity. Examples of these that we're going to go over are the Needleman-Wunsch, a classic algorithm, and the Smith-Waterman algorithm. Again, classic, came out decades ago. It's still used today in a lot of programs. Importantly, though, these can both be done by hand with a matrix. Starting with the global alignment, well, actually, let me take a step back because I want to mention that global alignment and local alignment are generalized terms. These are not necessarily only found in dynamic program. Any type of alignment you do can be categorized as global or local. That's just a little something to keep in mind when you're going forward. Most of the time, you're not going to have to choose between one or the other, but just keep in mind that there is a difference based on what your goals are. Now, starting with global alignment using dynamic program, we're going to have the Needleman-Wunsch algorithm. This came out in 1970. If you're interested in the history, or you want to see the paper, you can scan this QR code, which will take you to PubMed, or you can enter the PubMed ID. Just to give you a little history. Now, again, this can be done by hand with a matrix. And like we did with the dot plot and the dot matrix, we're going to try to align we're going to attempt to align these sequences, very simple sequences, by hand. This one's going to be a little more in depth than the dot matrix because we do have to do a little bit of mathematics, but it's really simple stuff, and you'll see that as we go through this. This is also where the coordinates become really, really important, because you can describe any cell of this matrix using the coordinate system, normally written something like table, subscript ij, or t, or table with the coordinates under here. So this cell would be t, or table 3, 1, column 3, row 1. That becomes important, just like in Battleship. That's become important when we start doing the math that we have coming up. So the Needleman-Wunsch and the Smith-Waterman algorithms have 3 steps. The first one is initialization, followed by matrix filling, and then backtracing. To go over the first part, we're going to fill in the top row and the far left column according to rules using the scoring scheme. Now, if you are familiar with alignments, you probably know about scoring schemes, but we're using just a very simple one here that will become, I hope, clear as we go through this matrix. A match, a mismatch, and a gap penalty is what you really need for a scoring scheme. We're just using a very simple positive 1 for a match, -2 for a mismatch, and a -2 for a gap penalty. After we introduce these algorithms, we're going to talk about the scoring schemes a little more in depth, but I want you to be able to understand how they work first before we get into what they actually do. So the first step is we have to do 3 calculations. These look really intimidating, but they're really not. And that's why I wanted to introduce the coordinate system. So every time you get to a cell, when you have to fill in the value, you have to do 3 calculations. The first one is you take the value that is ti minus 1, tj minus 1. All this denotes is, say we're looking at this cell. It's saying you take the value from a cell that is I minus 1, so over to the left 1, and j minus 1, up 1 row. So this cell. So this is just the coordinate system to orientate you on where you should take the value from. So first, if you were filling in this cell, you would take this value for this term, and then you would add this freaking looking thing, which is really just a fancy way of saying, is it a match or a mismatch? Again, using this as an example, we would have a T and an A. That's a mismatch. So we would put a -2. So all you do here is a 0 and then a -2. So 0 plus -2, -2. And that's the value you would put in the cell. Well, depending on what's the match, this will become clear. The second equation you have to do is ti minus 1j plus the gap penalty. Again, very technical looking term. It's just a fancy way of saying take your cell and move to the left 1 column and take that value. The j is the same, so you don't move up a row, so you just take that value. And then you add the gap penalty. And then the third is kind of the inverse of this. It's tij minus 1. So you stay on the same column, but you move up 1. So you can see the pattern in these 3 calculations. You take this value, and then this value, and then this value, and you do 3 separate calculations. For the one above and the one beside it, you just add your gap penalty. And what you finally do is take the highest score of this, like numerically. If you have a 0 and a -2 and a -2, you put the 0. We're going to practice this by filling in this matrix a little slowly, so hopefully it becomes clear. Starting with the first cell here. And keep in mind, again, we're still on the first initialization step, which means we need to fill in the first row and then the first column. So starting here, we just go through our calculations as I just talked about. Here, our first calculation, we ti minus 1, so we move left to a column, and j minus 1, we move up a row. There's no value here, though, so we can't actually calculate this. So we throw out that equation. But we can get the second equation, because that is I minus 1, where you move to the left, but you stay on the same row. So that's your gap penalty. So that's going to be a -2 here. And then the third term, again, is where you go ti j minus 1. You move up 1 row and take that value. But since we don't have a value here, we can't do this equation or this equation. So we only have this value to take, -2. So we add a -2 to this cell, and we add an arrow pointing at what value we took. We'll keep going this, and we'll see a variety of examples as we fill in this table. Now we move over 1. We go across the row first. And now we have the same thing. The first equation where we move to the left, I minus 1, and move up, j minus 1. We don't have a value there. We can't calculate that. So the second one the second equation we're looking at, we move just I minus 1. So move to this cell, and we add the gap penalty, -2. So that's -4. And then here we have, again, where we move up, but there's no value there, so we can throw that equation out. And we add a -4. So what you can probably see is there's going to be a pattern as you fill in the first row of your matrix. So really, you're just adding the gap penalty as you move over and add an arrow pointing towards the left. The same thing is going to be true when you do the first column in the initialization step. You can't fill in this cell or the first equation because you move over one, you're off the table again. The second one you can't do because, again, you're moving to the left. It's I minus 1j on your coordinate system, so you can't move there. Excuse me. And then, but finally, you can move up. So we move up j minus 1, and we add the -2 to get a -2. And you just repeat this process as you go down the column. This is a separate step because you generally moving forward, we're going to go across rows. But for the initialization step, you need to fill in your first column and your first row. This represents places where you have no matches between your top sequence and your side sequence. But once you're done with that, you can move on to matrix filling, and then you start here and you work your way across. But importantly, you do the same calculations that you did for the initialization steps. And now you can actually do all three equations. So starting here, we have to take the first equation, I minus 1, j minus 1. So we move over and up. We take this value and we add the match or mismatch penalty. We see we have a t here and an a here. That's a mismatch. So we take the mismatch value and add it here. So we have 0 plus -2. We have -2. Now we also need to calculate the other two, or use the other two equations to fill out this calculation, where we move first over one row, take that -2, and we add our gap penalty for this equation, second equation. That gives us a -4. And then the third equation, we take ti and t and j minus 1, and we have a -2. And we add, again, the gap penalty to -4. Now that you have three filled out equations, you just take the max value. In this case, it's -2, since that's higher than both -4 and -4. And importantly, you add an arrow pointing towards which cell you used. In this case, we used the 0 to get the value, so we point here. And you just move along the rows doing the same thing. I'm not going to belabor this point too much, but you move to the next cell and do the same thing. In this case, we have -2 for the first equation plus the mismatch because t and a do not match. So we have a -4. The next equation, we have a t I minus 1, so we move left. We have a j with the gap penalty, so -2 plus -2, -4. And now at the bottom, we have a -4 because we take the top value, I j minus 1, and add the gap penalty. So I hope it's becoming clear the pattern that's going on here. And since we have two values that are equal, we just put arrows pointing towards each cell. And we just keep moving on. We have the same here, since we have no match here and no match here. We just go through these same three calculations. We take the two highest ones, or the highest ones, and we put arrows towards where they go. Now, this is where it gets a little more interesting because now we actually have a match. So when we go through the first equation, we move left and up. We take the -6 and we add the matching value instead of the mismatch because A matches A. Now we have a -5 compared to a -8 for when we move just horizontally, and just a 10 when we move vertically. So we add our -5 value here. And this creates basically a route through the matrix that you'll see becomes important as you finish filling in the matrix. So when you get to here, you now just move down to the next row. And I'm not going to fill out all of these because this is getting kind of redundant. But once you fill out the entire row with all of your arrows, you're done with the second step, matrix filling. After this, and once all your arrows are in place and you're doing a global alignment, you move to the step 3, back tracing. For this, you follow the arrows. This is why the arrows were important. You follow the arrows from the bottom right to the top left. I've highlighted here how you can follow them. In some cases, you'll have multiple paths, and we'll show you how to deal with those later. But in this case, I just gave us a single path through from the bottom right to the top left. And from that, you can build an alignment. because you can use this information to figure out what aligns with what, basically. So we have a gap here with the A, but then we have the C. The C matches the G. This A matches this A. This T matches this T. This T matches this T. And then this A is at a gap, and this T is at a gap. So that's how we get our alignment from this very simple matrix. Once you have your alignment, you can do an alignment score by just taking the number of gaps, mismatches, and matches based on your scoring scheme and adding them up. This is important when you get to multiple types of alignments, because you, like I said, you may have multiple paths through your matrix, especially when you get big. This is a very simple, easy example. But when you get, 1,000 nucleotides versus 1,000 nucleotides in alignment, you can have multiple paths through your alignment, and then you can rank the paths through your alignment based on the highest score. This is a really crappy score. Generally, you would have a positive, but this is just for to show you how this works. Now, if you want to do a global alignment online, you don't really need, you can do these online, you don't need any software. The NCBI BLAST server actually has a way to do this exact algorithm, and so does the European Bioinformatics Institute as part of their Emboss suite. We're just gonna go over both of these. I'll see if I can click these without leaving my, yeah, I can't, I have to leave my presentation to click on these. We're gonna go to each of these so you can see. So I'm bringing this up and I will move it over. There we go. So if you go to just the BLAST server, the MCBI's regular BLAST server, and move down to their additional tools, you can see here we have a global align, which compares two sequences across their entire genome, Needleman-Wunsch. That's the algorithm we just used. It looks a lot like the BLAST interface, if you're used to that. We want to get, that is not for me. Now I am going to just add some basic sequences. So we can go here and just add two sequences. I'm going to add T4 distal tail fiber as an example, and a closely related tail fiber, or moderately related tail fiber from a phage called EP01. We'll talk about the additional parameters later, but you see you can do match and mismatch scores. The default is 1, -2 for the NCBI servers, I believe. Oh, I'm sorry, no, it's 2, -3. I had changed that earlier. And you can also change your gap calls. We're just going to leave these as default now because we'll talk about this in the future of this presentation, but really you're not going to be changing your default mismatch and match scores and gap calls very much. So once you have your sequences either pasted in or files uploaded, you just hit BLAST. It's not really a BLAST. Oh, did I mess up those files? Let me see. Let me double check and make sure these should be. Oh, I'm sorry. Whoopsie. I'm on the nucleotide. We're doing it with a protein. So let's try this again with a protein. And that's not, it says BLAST, but that's not really BLAST. It's calculating this algorithm. And you do get an interface that looks a lot like the BLAST viewer. You get % identity and your score. Here, when we talk about score, this is how that score is determined, just counting up the matches, the gaps, and the mismatches. You can see the graphic summary, but this only really includes your query and how much you have covered. It doesn't show you the matches and mismatches. To do that, you need to go to the actual alignment. Make sure my screen is sharing correctly. Yep. And we see we only have actually we actually have only 15 gaps, and a little over 1% gaps. We have positives and identities. So identities indicates how many amino acids are identical between our sequence 1 and our sequence 2. In this case, it's 78%. Our positives indicate similarity, and that is this value, the identities, % identity, plus amino acids that are similar. So if they have similar properties, they're both basic, they're both charged, they're both a nonpolar, something like that. They share similar sequences or similar properties. That is included under this graphic. So you can go down and look at your alignment here. You can see that you have mismatches. You just have gaps when you have mismatches, and you have positive when you have similar amino acids, and you have the actual amino acid when they match across. Now, there are some more things you can do with this. We talked about the dot plot earlier. This is how you can look at larger dot plots. And like I said, this is a great way to see where you really have mismatches or breaks between your two sequences. And I think that's all we're going to show for the global alignment, because there are some other things you can do, but we're going to look at this more when we look at the blast searches, because it uses a very similar interface. You can, using the alignment view up here, when you're looking at alignments, change how you view these two alignments between the subject and the query. So for example, these dots represent identical amino acids between the query and subject. And then these red amino acids here show the changes in your second sequence. And also do something similar with this. This is the query anchored view. This is better when you have multiple sequences to look at. But again, this interface is the same that is used for blast searches and the other searches we're going to talk about. Okay, so now that we've looked at that one, I'm going to bring you back to here. There's also, you can also do this through the European Bioinformatics Institute, and it's part of their molecular biology open software suite, the MBOS suite. It's going to be click on it now. No, it's not. But I'm going to click on this. And yeah, so if you go to this website, it brings up your MBOS tools. And you can go down to the needle. Needle is global alignment of two sequences using the Needleman-Wunsch algorithm. Just click this to launch it. Select either you have DNA or protein. We're going to look at the same sequences we did before. So we're going to have proteins. You can just copy one sequence up top, and then your second sequence right here. And you can change the output, but I like to leave it as pairs, but we'll go over some of these later if we have time. You can change the options, again, like gap penalties, and I don't think you can change, at least on this server, the match and mismatch penalty. You can only change the gap penalty. This should be pretty quick. So you see the interface here with the MBOS suite. While I'm doing that, I'm going to move these out of the way because they're annoying me a little bit. Okay, yeah, and see, this did not take long at all. You have some information up top in your output showing you some of the things that will run. It's kind of behind the scenes. You don't have to really worry about that if you're just doing global alignment. But these statistics are actually what you probably want to keep in these as well. If you change your gap penalty and you want to know what match and mismatch used. So we have identity 78.1%, exactly what we saw with the blast, and 86.4 for the similarity. Remember, in the NCBI server, we had the they call this positives. It's the same as similarity. And we had the same gaps, 15 with the same score. So you can see how they're comparable, and they use the same algorithm, and they have basically the same customization potential, but they give different outputs and they function a little bit different. So either one works. In this case, when you're looking at this output, the lines here indicate a positive match, like positive identity, and the dots represent a mismatch or a gap, and then double dots represent high similarity between the proteins or the amino acid. So, for example, like isoleucine and leucine, or what's another one? Like trying to think of an easy one, like arginine and lysine, but I don't see any of those in here right now. But I hope you get the point. You have your subject and your query and your subject, your sequence 1 and your sequence 2, and just they're aligned along with the residue number here on the right. You can open the alignment files and just download this or save this as an ALN file or a text and open it in another program if you need to. And you can save the submission details if you want to save that information. But not to belabor this point too much, because this is really the simplest of the algorithms we can do. There's also a third place to do this at a lab in the University of Michigan. They have a lot of tools here. We're probably not going to go over that because I believe we are already 45 minutes in and we've only gone over one algorithm. But we're going to be able to go faster now because what I want to show is how these all relate to one another. So that's the first algorithm. That's the global alignment. And this is how you can do it by hand. So I'm sure if you have a PI, I'm sure he's going to be happy to know that if the apocalypse ever happens, you can still do some work by hand. Congrats. You're welcome. The second type of algorithm we're going to look at is the local alignment, specifically the Smith-Waterman alignment. So this is the paper for the Smith-Waterman alignment. It came out in 1981. It came out in 1981, if you're curious to read the original paper. And it works a lot like the Needleman-Wunsch algorithm. It uses the same 3 steps. It uses a very similar matrix, and it uses the same types of calculations. The 2 biggest differences, one, negative values are set to 0, so you don't have negative values. And 2, you don't start at the bottom right when you're doing the backtrace. And we'll see that as we go forward. So again, you have to start with the initialization step where you move across the top row and across the first column by doing the same exact 3 calculations that are all going to be based on your match, mismatch, and gap penalty. We're going to use the same scoring scheme here as we did with the Needleman-Wunsch algorithm. And we're just going to go a little bit faster with this. I don't think we need to go through each cell individually as we did last time. So again, like I said, the biggest difference is you set negative values to 0. So once you have your -2 here, set it to a 0. And then you just move across. You would get a -2, -2, -2, and you just set these to 0 as you're doing your initialization. And you add the arrows. That's, again, important for the backtracing. And to see how this works here, now that we have all zeros, we actually have a -2 because we have a mismatch score of -2. For our first equation, we move over 1 and up 1, take the 0, plus the mismatch penalty because T and A do not match. That gives you a -2. The second equation is the gap penalty, 0 plus gap penalty, -2. The third equation is moving up, 0 plus -2, the gap penalty. That gives you a -2 all around, so you add arrows going to each cell. But since we're doing local alignment, we now make that 0. And we fill out the entire matrix according to that, according to those rules. And so you're only going to see positive values where you get a match or you get several matches in a row before going back to mismatches. So as you can see here, we have a lot of zeros and we have a few 1s, few 2s, and few 3. But we're done with the matrix filling part of the Waterman algorithm. But now we can do the backtracing. It's just like with the Needleman-Wunsch global alignment algorithm, except instead of starting at the bottom right, as I briefly mentioned before, and going to the top left, we start at the highest value and we keep going until we get to a 0. Like so. And this will give us a local alignment. So we're not attempting to align the entirety of these 2 sequences. We're just attempting to align the places that have high similarity between the 2 algorithms. So let me see that. Okay, so that gives us our local alignment, which again we can score the way we do with our global alignment. The difference is we're not going to be scoring like all the way across and including the gaps after the alignment because we're just focused on pieces that align. But you can score this the same way as you did with the global algorithms. And if you had multiple places to start, you would start at those, and you would, if you had multiple 3s to start at, you would start each of those and calculate the alignment scores to find the best alignments. And you can represent all of them or list the best ones. In this case, you'd have, match, match, match. Where you had gaps, you have a value of 3. So your alignment score is a 3 here. And that's really it for the dynamic programming. We went over it pretty fast. There's a lot to these algorithms that are still used in a lot of software today, but these are the basics and these are how they kind of work on an intuitive level. So I hope that helps you a little bit when you're looking at the dynamic or the pairwise alignments that you can do. We're not going to go over the Smith-Waterman algorithm like hands-on because it works the same way as the Needleman-Wunsch and it has the same output. So they work very similar. It's just whether or not you take regions of similarity or the entire sequence and align them. But there is a problem with these alignments. These alignments are so-called exact methods, which means they give you the exact perfect answer each time. The problem is they can be very computationally intensive, because the time to compute is proportional to the length of the input of your query. And if you're doing two sequences, either the length of your other sequence, oh, whoops, I'm sorry, I don't know why I did that. The length of your second sequence, or when you're doing a database search, which has several sequences, the length of your database. This is a problem because what we looked at before was just a simple 5x5 matrix. So we have this little thing here where we have 25 cells we have to do calculations for. But if you double that and you have a 10 by 10 alignment, so you have 10 nucleotides on one axis and 10 nucleotides on the other axis, now you have 100 cells to fill out, and then 400, and it just keeps going up. It keeps going up as you keep adding length to each, either one sequence or the other. This becomes a problem when you start getting to use actual sequences, or you get long sequences, or you get multiple sequences you're comparing against, because the amount of calculations that have to happen just increases, not exponentially. I forget actually at what rate it increases, but it increases more than linearly. So it becomes not feasible to do these types of alignments. As the length of the sequences increase, the number of calculations drastically increases. This becomes a problem, as I said before, with a database search, because when you're searching a sequence against a database, what you're essentially doing is doing multiple pairwise alignments. So if you're going to use an exact method like the Needham-Wunsch or the Smith-Waterman algorithm, then you have to go and do basically each calculation. So you do a matrix for each sequence you have. So your query sequence against the first sequence, the second sequence, third sequence, fourth sequence, up to however many you have. And remember earlier we said there's 200 million sequences now in NCBI. So you can imagine just how exhaustive this can become when you're trying to use this dynamic programming. And that's why I have here, this is really the time to compute. It is proportional to the length of the input and length of the database, because essentially when you have multiple sequences, that second length is just the length of all your sequences added together, the sum of all your sequences. So what do we do about this? We do have a great tool to handle this. And one of the first to do this was called the Basic Local Alignment Tool, BLAST. I'm sure everyone's familiar with BLAST. I think BLAST is now a verb. I'm going to blast it. I'm going to blast this protein. But not many people, I feel like not everyone understands how this works on more of an intuitive level. So I'm hoping that going through this can help. This was released, in 1990. In the paper here, again, if you're curious about the history and want to read the original paper. And this is really the bioinformatician's best friend. We love BLAST. It's called a heuristic approach. Now, compared to exact methods, which give you the perfect answer and the same answer every time, a heuristic approach is kind of a shortcut. It's a very accurate approximation. It's not guaranteed to give you the same answer every time, as like the database changes or whatever, but it does give you a very, very accurate approximation for very, very quickly. So instead of waiting, however long to do the exact method, you can cut that time down to a fraction using a heuristic method like BLAST. Now, there's one thing you should know about BLAST, is that it makes one pretty big assumption that I don't know if it's talked about enough. It assumes that good matches have short sequences that are exact matches. This means that if you have, if you have two sequences you're comparing, they're both 1,000 base pairs long, it assumes that, depending on the algorithm you use, that a piece of that, either, like 8 nucleotides or 27 nucleotides, match, that there's no gaps, there's no mismatches, there's no changes between those two. So that's why it becomes harder to use as sequences diverge, and we have to modify it when sequences diverge a lot. So, as I said before, this is really designed for speed so that you can search huge databases in a viable manner. And we're going to go over how that works again, like we did with the dynamic programming approach. So imagine you have a specific sentence that you want to find in a book. I actually asked ChatGPT to write this for me. Again, it was a pretty good one. She was delighted to find a xylophone among the musical instruments at the garage sale. You can look and read a paragraph. you can, or this is a paragraph, so assume it's for a book. You can go and you can read every sentence in that paragraph one by one. This is analogous to using an exact method. You go through, you read the first sentence, you look at your other sentence, see if it matches. Nope. Look at your second, you read your second sentence, you read your first sentence, your sentence, does it match? No. Third sentence, so on and so forth. But what you could do to speed this up is to just scan the book for specific words. Say you wanted to scan for maybe a rare word like xylophone. Then what you're doing is you're just scanning until you find the word xylophone or all instances of xylophone in your book or your paragraph or whatever you're reading. And then you're just reading those sentences. So now I have xylophone. Okay, she was delighted to find a xylophone among the musical instruments at the garage sale. She was delighted to find a xylophone among the musical instruments at the garage sale. So now that's how we can match these quickly without having to read every single word in our book. And so this is analogous to how BLAST works. It searches for small pieces rather than searching for the whole thing that you're trying to match. So it has to go through every sequence one by one. It takes small pieces and it tries to find where those match. So, yeah, and then it extends from there from xylophone for the whole sentence. So the first step to BLAST is breaking up your sentence, your sequence into words. BLAST calls them words. They are very similar, or they're basically the same in terms of concept as k-mers. Basically, what you do is you take your sequence, say the example sequence we have here, ATTCA, and you break it up into 3 nucleotide words. The actual BLAST uses bigger words. It uses, I believe it's like 28 for megablast. But just for an example, we're going to show you how you break them up into 3 nucleotide words. So if you have ATTC, at the first position, you have ATT. At the second position, you have TTC. At the third position, you have TCA. And so now you've broken up your sequence into 3 smaller words. And even though this is a bigger, this has more searches you have to do, it actually is less computationally intensive to search for exact matches for these against the database rather than this against the database with non-exact matches. These words are sometimes called k-tuples or k-mers. It's kind of a nebulous idea. K-mers is more of a general term, but when you hear the phrase k-mers, they're really talking about doing the same thing here, is taking a sequence and breaking it up into chunks. If you have a 7-mer, it breaks it up into 7 either nucleotide or amino acid chunks. A 10-mer, 10 chunks, or 10 nucleotide or 10 protein chunks. So just in the future for reference. But when they're talking about BLAST, they generally like to use the term words instead of k-mers. Yeah, like I said, this is an example of 3-mers. BLAST-N uses 11 mers, megablast uses 28 mers, and BLAST-P uses 5 mers. Now, once you have your words, you can search it against a longer sequence very easily. So here's the sequence we're going to search it against. Well, I don't have to read out, you can see it. And you can easily see the matches here and where they go to. So that's the first step, seeding. So once you've seeded your alignment, you know where short pieces of your query sequence match to, just like in our example of reading the book. This is like finding xylophone. And these are called, with BLAST, high-scoring segment pairs, or HSPs or HSSPs. This is an important concept if you're working with BLAST, or if you ever hear this, this is what it means. It means the seeding step where you have your words from your sequence that match pieces of either sequences in the database or just another sequence. Now, once you have the seeding done, you can convert this to a matrix. I didn't put all of the seeds that we have here because it gets a little clunky. I just put 3 to show you how it works. Now we can convert this into a matrix, and once we have this, we just do a Smith-Waterman algorithm. That's the next step. That's called extension. And what we're doing is we're really just like we did with the word xylophone. We're reading the sentence, we're extending out to the left and to the right of the seed and scoring the matches with a local alignment. So I'm not going to go over this since we did talk about how a Smith-Waterman algorithm is done. But as you can see, you start to see where it matches. So we have a match here with the seed. Does it match here? Well, no, that's a gap. That's off. What about here? A, C? No, that's not a match either. A, T, that's not a match. So again, the C, we have an AA here. We do have a match. And then here we have 2 matches, a C and an A. So we have a good alignment here. And we're going to score this. This is just a Smith-Waterman algorithm. And from here, we can pull out, oh, I'm sorry, this is called the extension step. So we have the seeding and the extension step. And then we can pull these out to make alignments. So now we have these 3 different alignment here that have quickly found matches to our sequence and performed a local alignment on them. And now we can store them to figure out which one is the best alignment. So, you're just, since it's a local alignment, you're just going to reach 0 instead of going all the way to the end. And so you're just storing the matches or mismatches within your block that you've aligned. In this case, of course, it's easy to see by eye, but when you're working with bigger sequences, this step becomes important and much more valuable when you're looking at how to do these. Okay, so when we're looking at just basic BLAST programs like DNA BLAST or BLAST-P, we have BLAST-N, which takes your DNA input, performs one search against a DNA database. This is like the first one, and this is the simplest one. This is good for moderately similar sequences. They then introduced something called MegaBLAST. This searches a DNA against a DNA database, but this is more geared towards highly similar sequences. So what it looks for is sequences that are more similar in a faster method. So basically it increases the stringency. Like I said before, I believe it was 28 mers that it uses as seeds instead of 11 mers like BLAST-N does. So you're going to get fewer seeds, but they need to be more similar. This is the default BLAST program. So if you do BLAST any proteins, you're probably going to be using this unless you change some parameters or tell it to do something else. And then on the protein side, we have BLAST-P, and that takes a protein input, does a single search against a protein database. There are also some more specialized BLAST searches that use the same method, but do additional searches. The first one is the BLAST-X. So this takes a DNA input and it translates all 3 forward frames and all 3 reverse frames. And then it searches each of those frames against a protein database. So as you can imagine, this takes longer because you have to do 6 protein searches against a protein database compared to BLAST-N, MegaBLAST, or BLAST-P. There's also kind of the inverse of this called T-BLAST-N. That's where you take a protein input and you take a DNA database that has been translated in all 3 frames. So you take a DNA database and you translate all 3 forward frames and all 3 reverse frames, and then you search that protein against each of those frames. And then finally, of your basic searches, you have the T-BLAST-X. That searches a DNA input against a DNA database, but it takes your query, your input, and it takes the 3 forward frames and the 3 reverse frames, and it searches those against a DNA database where the 3 forward reading frames and the 3 reverse reading frames have been translated. And so this is actually the most sensitive of them all. It's really good for finding matches, but it also takes the longest because, as you can imagine, you're doing more and more searches as you go up. Now, speaking of statistics of BLAST, we're not going to spend a lot of time talking about how these are derived or where they come from, but there are 2 values you should be aware of. The first one is the expected value, or E value. This has an equation that can be derived from, and it's based on the size of the database you're searching. But really, for your purposes, for most people's purposes, you just want to think about this as what is the probability that this alignment happened by chance. So this value is going to decrease as it increases as the size of your database increases, because if the size of your database increases, the chance that it happened by chance, that you have an alignment by chance, goes up just because you have more combinations. But it's still really easy to get good matches, so you don't have to worry about this too much. All you need to know is that if you do get an E value that is, close to 0 or above 0, or I'm sorry, higher than close to 1 or above 1, then it's not going to be a good match. Anecdotally, there was an instance with the COVID pandemic where, I don't know if you guys remember, but someone somewhere decided to align the draft COVID genome against HIV. I think they used BLAST to do it too. And the expected value was something that was actually really high. Like it wasn't a significant value. I can't remember what it was. But the person doing this didn't realize that. And so they posted it on a forum, and then it went viral. People saying that it was HIV in the COVID genome. And There wasn't. And that's because the person doing this didn't understand the expected value. The other score to be aware of is the bit score. This is a log2 scaled and normalized raw score. Recall how we talked back here. Let me start bringing that up. Right here about scoring these alignments. This is that's a raw score. The bit score takes that raw score and normalizes it and puts it on a log2 scale. And this tells us how good our alignment is, regardless of what are the chances of it happening at random. So if you have a bunch of scores that are all significant, you can rank them by the bit score to figure out what the actual best alignment is. So the e-value doesn't necessarily the best alignment, but the bit score should be the better alignment. Okay, so now we got another hands-on portion where we're going to go through some of the things we can do with Blast in the slideshow and bring that up. I'm going to put it over here so you can see the link, or if you want to use the QR code. So I'm going to bring it up this way. So again, it brings us back to the same interface that we had before. We're going to start with a nucleotide blast because that's the simplest. And we'll look at the interface a little bit. So we can go here, and I actually do not have, I don't think I have a nucleotide sequence ready. Let me grab a nucleotide sequence from somewhere, and I will use that as an option. But first, we'll start with the blast P since I do have those ready. So let's do this. We're going to just, instead of uploading, which we can do, and what we did before, I'm just going to copy this, hit C, and you can paste it as a text. And we can look at our options. So with the blast in the nucleotide, we can go down here and check our different algorithms we talked about. We didn't talk about the discontinuous blast, can't say that, but we did talk about the blast in and the mega blast. The mega blast is going to be the standard default one we use. We also have a lot of options here. When you add your query, you can tell it what you want to search against. So if you want to search against the full sequence, you can just leave this blank. But if you have certain regions, you can tell it where you want to start, maybe the 10th residue or the 10th nucleotide or whatever, and where you want it to end. I generally don't use these very often, but they can be helpful. You can either paste your sequence or you can upload your sequence. You can add a job title, so it's easier to find your specific search through your files if you're logged in. You can also align two sequences using Blast. This is a lot like the Needleman-Wunsch algorithm we looked at before, except it uses Blast to align to. It uses whatever Blast algorithm you tell it to use to align two sequences, which is completely fine. Essentially, that's all Blast does, is align two sequences. It's just you don't need a heuristic method like Blast to align two sequences, especially if they're two short sequences. Works better, and you have more options to customize if you use something like a Needleman-Wunsch. But it's completely acceptable to use a Blast algorithm to align two sequences, because that's essentially what it's doing when it's searching the database. It's just optimized to search databases. But we don't want that right now. Another good thing about the Blast, the Blast server, is that it has a lot of information. You can just click on these little question marks if you have questions about what each thing does. And oftentimes you can hit more and it gives you more information you can follow up on. We're not going to go over all this. This is something you can do if you need to. See what else. You can also choose the database you want to search against. For most purposes, you're just going to use the standard databases, and you can read guides about which database which database is best for what. We're not going to focus too much on database today because they're pretty simple to understand. But the default one is the nucleotide collection if you're searching for nucleotides, or if you're searching for BLAST-P, the non-redundant protein sequences. These non-redundant databases just take out sequences that are 100% identical. And so it merges them into one entry. So if you have, say, 5 proteins from 5 different organisms or 5 different strains of the same organism that are identical in a database, it takes all of those sequences and it collapses them into a single sequence. But you can go from that sequence and figure out which ones are identical to it. And I'll show you how to do that from the results page. So we have, going back here, you can also limit this to, you can also limit this to organisms. So if, for example, you only want to search E. coli, you can just start typing it and it will give you the tax ID code. For this example, this is Escherichia, so this is the genus. If you want to do the actual species, you can add this, which is the next tax ID. So what this will do is if I leave it here and add, whatever my sequence is, ATGC, whatever, this is only going to search against the E. coli in this database. It's only going to search your sequence against the E. coli in this database. This can be useful if you know what you're looking for and you're trying to find homologs in a certain species. But you can also hit exclude here and exclude E. coli. So if you know you have a sequence that is in E. coli, for example, and you want to exclude these types of organisms, or this species specifically, you can just hit exclude here, and now it will search the database for everything except E. coli. And this can be useful when you want to trace kind of like the evolutionary history of the protein, or if you want to see if it's found in another organism. You can also add, up to 20 different ones. So if you want to search a collection of genuses or species or any level of taxonomy, you can do that here. But generally, you just leave this blank and it's going to give you the best results if you don't. You can exclude certain things, but this isn't really big a problem if you choose the right database, just like you would want to exclude uncultured environmental sample sequences. But that's not generally a problem when you're working with defaults. This is more if you're using some of these more specialty or bigger databases, like maybe maybe if you're searching for like sequence read archives, which searches your query against reads that have been submitted to the SRA database. So yeah, we're going to start with BLAST P since I have that sequence ready. I should still have that on my clipboard. Yep. Okay, so this is the T4 distal tail fiber. If we want to do a BLAST P, this is the default. We have the same options here with the different databases, but we're going to leave these as the defaults. You can also exclude organisms, but we're not going to do that right now. And we're not going to add a job title. We're just going to do this as is. But if you go down to the bottom, you can see additional algorithm parameters. And this is where you can change some of the stuff I talked about before. With protein, we have some things we haven't talked about yet, like the matrix score. We're going to talk about that next in our presentation. We do have the gap cost, which we saw in our example was -2. In this case, the existence is a 11, and then extending that gap costing 1. And then we have some things we're not really going to talk about because they're more advanced, like the complexity filters. You shouldn't have to worry about this unless you get more advanced. The things you will worry about are the max target sequences. If you add 100 and you get back all very similar sequences, but you want to be more divergent, you can just keep going up until you have more and more sequences and then go down more or less. You can change the word size. Remember when we were talking about how BLAST works, we broke our sequence up into words. In this case, they're basically like K-mers, but in this case, the default is 5 for BLAST P. So we can leave that there, but you can also decrease it as little as 2, which is good for, you'd want to decrease it as the proteins you're searching for become more divergent because there's going to be more differences between them. It's more likely that you're not going to find 5 amino acids in a row that one hasn't changed. Because recall that BLAST requires in the seeding step for the seed, the words, to have exact matches, no gaps, no mismatches. So we're going to start with this, with the BLAST P, and just hit BLAST. This shouldn't take very long. And while this is working, I am going to pull up a sequence, I believe, or a nucleotide sequence, because I only have my protein sequences to look against for these examples. I don't know why I did that. Normally, BLAST P, BLAST N, and MegaBLAST do not take that long, so you can get them started and just wait a little bit. But some of the other algorithms do take longer, and especially when you start doing multiple sequence alignments, which we still haven't gotten to, you will see that the intensity and the amount of time it takes to compute these things go up a lot. Let's see. Okay, now that we have that done, I now have a nucleotide sequence. So again, we have the interface that looks a lot like the interface we have when we did the Needleman Wunsch algorithm. We have a summary here of the hits we have from the database. We have the scientific name of the hit. We have the max score and the total score. This is going to be the alignment scores we talked about. We have the query coverage. This just indicates how much of your query is covered by the hit that you have. So if you're looking for close matches, you would want 100%. But if you're looking at something that has a very common domain, you might find something with a smaller query hit, like 50%, where it still covers the domain. We have the e-value. Recall we talked about that. What are the chances that this alignment happened by accident or happened at random? And in this case, you can see they're all 0. We have the percent identity from 100%, dropping quickly down into 70, 80%, and the length of the alignment. We also have the accession numbers here to the side with the sequence. So if you want to see the sequence that aligned to, you can just click that, and it should bring up. Yep, 100% match was the long tail fiber distal subunit. You can get more information about this from NCBI. I'm not going to go over a lot of this, but there is a lot of things you can do. You can identify a conserved domain. You can highlight sequence features. If there is a protein structure, it's available here. You can just click on that to go to it and visualize it. So this can be really helpful. And it shows you some domains under this graphic and shows you what chemical and non-standard residues are found in the structure. It also lists, this is very helpful if you're looking for more information about your protein. It also lists papers that have talked about this protein specifically. Here to the side. And I think that's about all we're going to talk about. There are some related information here, but if you get curious, you can go through this yourself with your own protein. So we can go back here and go back to the graphic summary. This again shows you more of the query coverage, and it also gives you what protein domain hits. We're going to talk about how these conserved domains are searched for in just a bit, but we have our query coverage here, and you can see how most of the proteins that hit cover 100% the entire length of your query, but some of the more distantly related ones start losing the first part. And interestingly, if you look at this, you have a collar domain. You can click on this to bring up the domain search. Yeah, this is a phage tail collar domain. And so this is also as helpful when you're looking for a specific domain in your protein. You can see where this domain is on your query and kind of estimate it. This one looks like it's around 800. And so around this area, and see which of these proteins actually have that same domain. You can look at the individual alignments. This is what we looked at before. It has the same information that the global alignment had. We have the percent identity, we have the percent similarity, and we have the gaps. And also recall how I told you how the non-redundant databases collapse proteins that are 100% identical to one another into a single node and searches against that node. This is where you can go and look at the different iterations of that, which are in that node. So, we were looking at this T4 distal subunit, tail fiber distal subunit, but there are also other sequence IDs that include something similar or an identical protein to it in different sequences. So you can go here under see more titles and even go to see all identical proteins to see which proteins are found to be identical to the one that you got the hit for. There we go. And so you can go through and find any more information about these proteins here. This is very useful when you're looking for maybe really closely related species or strains that have the same identical protein. If you're not finding it in your blast search, you probably can find it under identical proteins. And we can also go to taxonomy. So, well, before we go to taxonomy, I wanted to point out the alignment view we talked about in the global alignment. You can change how you view this, and it may be easier to view in different ways. So in this dots for identity, the dots mean that it is the same as your query. So it's easy to highlight the differences, easier to see the differences between your hit and your query. And we can also do a query anchored. This is almost like a multiple sequence alignment. So you have all your hits going here, and you can see where the changes are compared to your query. The dots represent identity or identical residues, whereas the capital letters represent amino acids that have changed. And this only shows the first 60, but you can change that to 150 max to look at these proteins. The other ones are kind of the same. It's just this is a little different. This is a little harder to see where the changes are, but you can align it this way. Or you can do the flat query anchored, which is, for our purposes, the same as the query anchored. Then you go to taxonomy. If you want to find hits in a certain species or a certain genus, this is where taxonomy comes in handy. The lineage reports show you how many hits you have in the different lineages, including the max score of these, or the average score of the hits. And you see as they go down, as they get more divergent. And you can also do it under organism. So this looks at each organism and what your hits are for that organism. This is more useful probably for bacteria than it is for phage, because we have so many divergent organisms for phage. You start to see that when you get down to, like, let's see, I guess it's not bacteria, it's not on here. But lastly, and probably the most useful one I like, is the taxonomy report, because this in here you can easily collapse the trees that you don't want to see. So if I don't want to see the viruses, I want to see where this tail fiber, where I have hits for this tail fiber in bacteria. So I just collapse the viruses and look at the Enterobacteriaceae. And we see we have hits in Shigella, E. coli, and Salmonella. So if you're looking for your tail fiber in certain species, this is a good way to find those hits. And you can click on them to bring up the more information about the hits you have. It's not really loading. There are a few other interesting things you can do. You can do a distance tree results. This just creates a fast tree of the hits you have for your query and how they're related to one another. This is not something most people are going to work with, but this is useful if you're looking, again, at the landscape for your protein. And you also have multiple alignment and multiple sequence alignment viewer. We're going to talk about those when we actually talk about multiple sequence alignments next. But for now, we're actually going to go and look at the blast in, because we see similar things with the blast in. So I'm just going to copy the distal tail fiber nucleotide sequence, leave everything the same for blast in, for megablast defaults, and we're going to go forward. While that's going, we're going to open back up blast. No, which one is blast? Go back to nucleotide blast and look at the algorithm parameters. You may have noticed that when looking at the protein parameters, we didn't have a match and mismatch score. We do for blast. We have a gas cost as well. And by default, we also have low complexity filters, but those aren't something we're going to talk about today. Those are a little more nuanced than we're going to talk about. But basically what those do is, think of it as filtering out regions that are expected to happen often by chance. So if you were looking at, we're talking about our word example, where you're talking about finding those xylophone in a book, it may be like masking out the word the, because the is found a lot, and it's not very useful to get hits for the word the and read every sentence that has the word the in it. Okay, so now this is done. This looks a lot like what you have for the protein blast. You still have the same, you have your scores, you have your query coverage, you have your e-values, your percent identity, and your length. So what you see here is that you get hits for both genomes and for pieces of genomes. Like this is a piece of T4 tail fiber, gene 35, 36, and 37. We're looking at gene 37, that's the distal tail fiber. And so bringing that up brings up your, brings up this select sequence, not the full sequence, but it gives you sequence for these few proteins from T4. Whereas if you go to the complete genome hit and click the accession number, this is going to bring up the complete genome for T4. So this is one of the drawbacks to using nucleotide blast instead of protein blast. The protein blast gives you specifically your protein, where the nucleotide blast directs you to the sequence that contains your nucleotide sequence. But other than that, things act the same. You get your graphic summary, which shows you your query, and this has different scores, so it shows you the average alignment score in that region. So 200 is good, so red is good. but it starts to get lower and lower in some places as you get more divergent, and you have a lot of gaps in your query. Okay, again, look at the alignments. You can change your alignment views. Like we can look at the query anchored. If you want to look at all of them and see what the differences are. Okay, so I think we've pretty much exhausted some of the basic options for BLAST. I will link you to some more tutorials later, because BLAST has a lot of really good tutorials on YouTube that you can follow along with. They're much more in depth than what we're doing here. They talk a lot more about what's going on under the hood. And so those will be next. Oh, yeah, I have it here. I made a tiny URL for this because it's kind of long, but if you just find the National Library of Medicine on YouTube, you can find their tutorials there. If you have a phone, you can scan this QR code, and that'll take you, or hopefully should take you, to this BLAST tutorial playlist on YouTube. Okay. Now we're going to talk about scoring schemes a little bit before we move on to multiple sequence alignments. So recall in all of our examples before, we've shown a mismatch of +1, a match of +1, a mismatch of -2, and a gap penalty of 2 for all of our examples. In the Needleman-Wunsch algorithm, the default is generally 2 for a match, -3 for a mismatch. This is actually for nucleotides. I hadn't mentioned this before because it gets a little more complicated. But with nucleotides, it's fine to use a so-called binary scoring system where you have a score for matching and a flat standard score for mismatching. This is because there's only 4 possibilities at any position. There's roughly the same chance for each base going to another base. Obviously, if you're familiar with evolution, you probably know that this is not exactly true at any one position, because something like a T to an A may be favored if it's a certain place in the codon. The problem is it depends on where that nucleotide is in the codon. It depends on if it's in a coding region. It depends on if that coding region that it's in is important. It depends on a lot of factors. So all of that kind of equals out to where you can roughly say that they are equal enough that you can use a binary match and mismatch system. When you're deciding on a match and mixed match ratio, generally there are guidelines, but people don't really tend to follow them. They normally just use the default because it generally works well enough. But if you actually want to get an improved alignment score, what you can do is use the NCBI recommended ratios of match to mismatch ratio, or mismatch to mismatch score. So a 1/3, that is a +1 for a match, a -3 for a mismatch, is good for highly conserved sequences. A 1/2 ratio, we have for example, a +1 for a match and a -2 for a mismatch, which is what we had. That's good for 95% conserved regions. And a 1-to-1 ratio is good for less conserved regions, like a +1 for a match and a -1 for a mismatch. You can think of this as not wanting to penalize gaps or mismatches, because the further sequences get apart, the more mismatches you're going to have. And if you penalize them too much, they're going to start avoiding them at all costs, and it can really screw up your alignment. So you don't want your mismatch score to be too high and penalize mismatches too much, because then it's hard to find good alignments. But at the same time, you don't want to under penalize them, because if you do that, then you may have too many mismatches in your alignment, and you can struggle to find a good match. This is more on the theory side, though. In practice, you're really probably not going to be changing your match and mixed match score very much. I just wanted to point this out so you can understand that what we've been talking about before in this binary scoring system is really something that's seen on nucleotides, not on protein. And the reason that is because proteins are more complicated. Instead of 4 possibilities, you have 20. If we're not talking about exotic amino acids, you have 20 possibilities. And all of these amino acids have their own properties. So you have polar side chains, non-polar side chains, you have charged side chains, you have basic residues and acidic residues, you have size differences, and all this can factor into which mutations are likely to happen. For example, an arginine to a lysine may be more common than, say, an arginine to a tryptophan, because lysine and arginine have similar side chain properties. And this goes back to what we talked about with our hierarchy of structures. When you're thinking about a protein on a 3-dimensional level, a protein needs a certain configuration, a certain shape to do its job. And that shape is based on the secondary structure, which is based on the primary structure. So this tertiary structure, this three-dimensional protein structure, requires interactions between the primary structure inside it. So if you can imagine, you have like, say, two domains that come together. Somewhere in there, you may need two interactions to happen, or a hydrogen bond to happen, or something, some kind of interaction here, or to bind to something so that this protein can keep the correct structure. And what that means is that when it mutates away from that and maybe misfolds the protein, that is selected against. Whereas having an amino acid that is similar to the one that was there before when it mutates, that may be not as ideal as the original amino acid, but it's not selected against as much. So that means scoring systems become much more complicated when we're dealing with proteins. So yeah, like I said, proteins are complicated. Now, the way we deal with this is something called substitution matrices, or under our scoring schemes. There are a few, but the two main ones you probably heard about and you see in a lot of programs are the so-called points accepted mutations, PAM, or BLOSSOM, and then the other one is BLOSSOM. So the PAM matrices were calculated, I think in 79, in a really interesting fashion by taking global alignments of related proteins. They took something like 70 or 71 protein groups that were very similar, and they aligned them together, and then they calculated the amount of change in these proteins over certain evolutionary distances. The first PAM was PAM1. So using these proteins, they modeled evolutionary distance, and they set that distance to roughly a 1% divergence, or one change per 100 amino acids to be expected. And then they calculated the probability that certain amino acids will go to certain amino acids. So what is the probability that an arginine becomes a lysine, or arginine becomes a histine, or arginine becomes a tryptophan? And they made matrices with these. You're probably familiar with, if you've seen PAM in some of these programs, the software programs, and you'll see PAM with like PAM1, PAM120, PAM250. PAM250 is probably the most popular because it works really well, or works better for domain searches. So PAM250 is probably the one that people see a lot. This is an evolutionary distance where you expect 250 chains per 100 amino acids. That sounds like a lot, like a protein would be completely different. But keep in mind that when we're talking about this, it gets a little convoluted, but you have changes that can happen multiple times at the same residue over this distance. So over this evolutionary distance, proteins are still expected to be about 20% identical. Now, the interesting thing about PAM, or one thing you should know about PAMs, are that they're all extrapolated from PAM1. So once that first probability was calculated, they just multiplied it by itself, because you can do this with maths. It multiplied by itself to get increased evolutionary distance. So 2 mutations per 100 amino acids. To get PAM3, you multiply PAM1 times PAM1 times PAM1. So PAM1 cubed. Excuse me. And so that keeps going up and up and up until you get PAM250, where it's PAM1 times itself 250 times. Now, what this looks like when we're looking at the substitution matrix is a matrix that looks like this, where you have amino acids going on the columns and the rows, and it's just showing how these relate to each other in this heat map. These values are log odds ratios. That's a fancy way of saying it's a log transform of probability. So it's a way of standardizing it based on what the expected amount of change is. For 0, that means under this condition, PAM250, where you have a 0, that means that the number of substitutions you get where you see, say, a cysteine go to a serine, 0, that means you get the number you expected. So it's neutral. This is a neutral mutation. You go from cysteine to serine, or serine to cysteine, and you get that as many times as you expect to get that over that distance. Now, when it's positive, you actually get that change more than you expect. And when it's negative, you get that less than expected. So for example, when we're talking about, I probably should show this. Yeah, this is just what I was saying. Positive numbers indicate substitutions that happen more than expected. Zeros are substitutions that are neutral and they happen as often as you expect. And negative numbers are substitutions that happen less than you expect. So what this looks like when we're looking at like related proteins, we have like the histine, the lysine, and the arginine here, all basic charged amino acids. We can find them on the matrix here and look at their values. So this tells us that an arginine to arginine is by far the most likely thing to happen. But an arginine to lysine happens a lot more than neutral. It's not a zero. A zero would be neutral. So it happens actually a lot more than expected. And it's because lysine and arginine are similar, meaning that an arginine mutation to lysine or a lysine mutation to arginine can happen without disrupting the protein too much, basically. And so you can see that same with arginine and histine and yeah, any of them that are similar like that. So I know this is a little convoluted and maybe a little hard to wrap your head around this. The only thing you really need to know about the scoring matrix is that it's just a logarithmic transformation of probability, which is the same for BLOSSOM, block substitution matrices. This came out much later than the PAM. And the PAM, while it was done by hand and it was done really well, it really doesn't have that many proteins that it uses. It only uses, I forgot how many changes. It was 1,572 changes, I think. Well, BLOSSOM is better because it uses more proteins to calculate basically the same type of matrix. The biggest difference is that it derives these matrices, these probability matrices, from local alignments of distantly related proteins rather than closely related proteins. And it's always based on observed data, not extrapolated like the points accepted mutations are. So this is just an example of what it looks like. As you can tell, they're very similar. They look very similar. They just have different values because the probabilities are different based on how they calculated it. I do have some information about the differences between these two. For example, like I said, the PAM is based on global alignments, where the BLOSSOM is based on local alignments from divergent sequences. The PAM1 is derived from sequences that have diverged by 1%. That is, their evolutionary distance is 1%. You have the BLOSSOM 62, which is derived from sequences that are sharing 62% identity. And then it talks about how, and here we have how they differ by how they're calculated. The PAMs were extrapolated from, the PAM matrices are all extrapolated from PAM1, whereas the blocks are directly observed frequencies. One thing to note is that as PAM gets higher in number, we have it here, yeah, as PAM gets higher in number, the evolutionary distance increases, so it's used more for proteins that are more distantly related as they branch apart. Where BLOSSOM is the opposite way. As BLOSSOM value decreases, the proteins used get more distantly apart to calculate these substitution matrices. So when do you use each of them? PAM is not used as much as BLOSSOM. PAM is used for sequences that are believed to have closer evolutionary relationships, whereas BLOSSOM is commonly used for many database tools, and it's found, specifically BLOSSOM 62 is found to be superior to most other BLOSSOM substitution matrices and PAMs. Now, if you wonder how this works, like if you're doing this substitution matrix by hand again, instead of using a match or mismatch score, you can use a substitution matrix. If you were keen, when I looked at the additional algorithm parameters on BLAST-P, you probably saw that BLOSSOM 62 is the default for match mismatch, and you can change that to different algorithms. Basically, what it means is that when you're doing this matrix, once you get to a place like, say, we're here, where we're doing these three calculations again, always these three calculations, where first you move to the left and then up, take the 0, and then you take this, then you have to calculate match or mismatch, and you get that score from this log transform substitution matrix. So in this case, we're looking at 5 because they match. Where if we were looking at, say, like valine and proline, if we saw a change here, then we would put, let's see, proline, valine to proline right here. So that's a -2. So that is our mismatch score. So I hope that clears up how we use substitution matrices for proteins and why we use substitution matrices for proteins where we use binary scoring systems for nucleotides. I'm not going to focus much on gap penalties because, to be quite honest, no one really changes their gap penalty. Generally speaking, most people don't want to change their gap penalty because you have to justify it. And if you don't move from default, you don't have to justify it. A gap penalty, all it does is assigns a score to to an open or extend a gap. And so you can penalize, or you can decide how much you want to penalize a gap in your alignment. Yeah, for example, here, if we have an alignment, you can penalize insertions or deletions in your alignment based on that score. And like I said, most people do not change the default score because they have to justify it. But there are some times you might want to justify it. For example, if your initial alignment has too many gaps or more gaps than you think it should have, you can increase your gap penalty score. So instead of, for our examples, we use -2 gap penalty, you can increase that to like a -4, and to try to bias the alignment away from gaps. Another time is if you're aligning regions that you know are more or less prone to insertions or deletions. So if you're aligning a region that is prone to insertions or deletions, then you can decrease your gap score so you don't overly penalize them. If you have a region that is more prone to insertions and deletions, or less prone to insertions and deletions, you might want to increase the gap score or the gap penalty to bias away from introducing gaps into your alignment. And so we've gone over dynamic programming algorithms and heuristic algorithms. I hope it has been clear so far. We only have the multiple sequence alignments and hidden Markov models left, and we have about 1 hour and 20 minutes, I believe. So I think we're making good time on this. So what we've talked about before are pairwise sequence alignments, and that is the alignment of 2 sequences. Those are pretty simple, and those can be done on a two-dimensional matrix. But when you start doing multiple sequence alignments, it gets really, really frustrating and computationally intensive, because every time you add a sequence, you need to add a dimension. So if you're aligning 3 sequences, instead of having just the front face of this, the 2-dimensional, you have a 3-dimensional. And you can do this. You can use the exact methods we used before, the dynamic programming, and you can place arrows in a 3-dimensional space like this, and you can calculate multiple sequence alignment by hand with 3 sequences. But this starts getting much more complex when you go above 3 sequences. Mathematically, you can still do that, but as you can imagine, the resources that takes and the time that takes increases as you add more sequences and as those sequences get bigger. So in order to get around this, people came up with some other heuristic methods. The first of which is, or the most popular, is the progressive multiple sequence alignment. And this uses pairwise alignment to progressively build a multiple sequence alignment. And these algorithms are still used, and I still use them myself. The first paper was out in 1987, so this is actually even before BLAST came out. And it's a really interesting paper to read. It follows similar steps. First, it starts with an all-by-all pairwise alignment. So when we talk about dynamic programming, it uses that, but in this case, say we have 5 sequences, it aligns each of these sequences by every other sequence. So basically, it does multiple pairwise alignments, and it figures out how closely they are related. The second step is it creates a distance matrix. A distance matrix is simply how closely related these 2 sequences are. And depending on the algorithm you use, you're going to change different multiple sequence alignment algorithms are going to use different programs and different methods for each of these steps. This is just like a general overview of how they work, because they all use very, very similar steps. They just slightly modify it. Some use dynamic programming, some use like hidden Markov models, some calculate the distance matrix trees in certain ways. So this is just a general overview of how they happen. But in progressive multiple sequence alignments, once you have your distance matrix, however you create this matrix and whatever scores you use, you then build what's called a guide tree. This is simply taking the sequences and attempting to build a tree to figure out how closely they are related. And this guide tree guides how your progressive alignment moves forward, which is your next step. So if you align sequences using a guide tree. So in this example, we have sequence 1 and sequence 2. Those were the most closely related. So this means that those are going to be our seeding pairs. So we start our multiple sequence alignment by aligning these 2 as if they were, just a pairwise alignment. So we're doing this, again, mostly dynamic programming. Some programs use non-dynamic programming, but you can use dynamic programming here to align these 2 sequences. Now, once you have those 2 aligned, you can add the next sequence, which is going to be more distantly related. And now you take the consensus of this and you add the consensus of the sequence 1 and 2, and you align sequence 3 to it as if you're doing a pairwise alignment. And as you probably guessed, you now move to sequence 4, and then you do the same thing, consensus versus sequence 4, to make this alignment. And then finally, the most distant sequence you added here. This is called a progressive multiple sequence alignment, and it's a heuristic approach, meaning that it is not exact and exhaustive. It doesn't guarantee to give you the best possible result, but it does give you a very accurate approximation most of the time. Typically, they use global alignment algorithms, so there are some modifications that can use global, but generally speaking, they use global alignment algorithms. And the biggest problem with them is they tend to propagate errors. So if an error is introduced early in this alignment, it will propagate throughout the alignment. So if you have a if you have a misaligned, say here, that error is going to propagate when you add the 4th sequence. And then it's going to propagate more when you add the 5th sequence. And the nature of progressive sequence alignment, multiple sequence alignment, means that you never go back and adjust these alignments. Once this alignment is done, it's set. You add the next sequence. You never go back and change this. And so, yeah, so that's a progressive alignment. The next type is iterative multiple sequence alignment. And as you can guess, if with this type, you never go back and correct anything, the iterative multiple sequence alignment, you do go back and correct and readjust things. So as you can imagine, those use more computational resources. The first paper that I know of came out in 1991, so only a few years after the progressive multiple sequence alignment. And this is an improvement in accuracy to the progressive. This is pretty easy to explain too, because it basically does, these types of algorithms basically do the same thing as the progressive sequence alignment. Except when you get to the end, depending on which type of iterative algorithm you use, it goes back and it corrects something it's done before and tries again. For example, I believe it's muscle uses a step where it cuts open the guide tree into 2 separate sequences. Then it aligns those independently as if they were different multiple sequence alignments. And it takes this realignment and then it merges them together based on the consensus. And then it compares them to see if the new alignment up here is better or worse than the old alignment. If it is, it then cuts the tree again and tries again. And it keeps going until you either tell it to stop or it reaches convergence where the score no longer improves. And so the idea here is that, remember I said with the progressive multiple sequence alignments, you propagate error. Once you have an error that's introduced early on, it can propagate and screw up your whole alignment afterwards. Well, these iterative multiple sequence alignments attempt to correct for that, or to at least mitigate that somewhat, but they're still considered heuristic approaches because they can't guarantee a perfect result. So yeah, they're typically more accurate than progressive, but also typically take longer and take more resources. I made a little table here just giving you some of the differences between progressive and iterative alignments. One thing to note too is that how you categorize one versus the other is a little, it's not so clear cut, because some of the progressive algorithms do have steps that correct and refine, and they refine themselves, but not truly iterative. So just know that sometimes you'll see an algorithm, and sometimes we will describe it as progressive, and sometimes they may describe it as iterative. Excuse me. Okay. So yeah, the progressive is less flexible. The iterative is more flexible and more accurate, but it's also slower. The iterative is generally good for medium to small alignments, where the progressive is better for medium to large alignments. Progressive is still good to use, especially if you have closely related sequences, like sequences all within the same species. Where the iterative is good for looking at more distant relationships. Yeah, I think I've gone over all this. The progressive algorithms can propagate the error. I really want to pound that in. And the iterative requires more memory and computational power. Some of the most well-known types of software are Clustal and Clustal Omega. It depends on, again, for example, how you classify these as iterative or progressive, because again, it's not super clear cut. For example, the Clustal Omega, it does incorporate a clustering algorithm that helps refine it, and so that can be considered iterative. But some people still classify it as progressive. MAFT, which I really like, is a really good algorithm for very fast multiple sequence alignments, and it works really well if you have a lot of sequences, like up in the input to the hundreds. If you use some of their methods that use fast Fourier transforms, which don't even worry about what that is. It's a mathematical concept, but their progressive algorithms are really good at using are really good at putting together multiple sequence alignments of longer sequences and mini sequences. But it's not as accurate as some of their iterative algorithms, which can be really slow. But some of the algorithms they have are actually really, really accurate. One that I do like using is tcoffee as well. Tcoffee is a progressive algorithm, but it does have some refinement steps and some ways that it improves upon the basic progressive algorithm. I've had good luck with getting good alignments on kind of a larger scale, the medium to large scale. And probably one of the most well-used ones is Muscle. Muscle is a really fast iterative algorithm that does really well for larger data sets, despite being an iterative algorithm. And so we're going to look at that one now, actually. The Muscle algorithm can be found here. I'm going to leave this up for a second in case you want to get the PubMed ID or look at the website. Again, this is from the European Bioinformatics Institute under one of their software. Oops, let me do that. Meant to do this. Okay, so this interface looks a lot like what we saw with Needham and Wunsch, because they both are from the European Bioinformatics Institute, which has a lot of good tools. If we have time, we might look at some of the broader tools. And they also have several for multiple sequence alignments. You can go here to tools /MSA, and you can see we have the cluster omega that we talked about. We didn't talk about cons or Klein, but those are pretty good. MAFT, I really like MAFT for the larger alignments. We can do Muscle here. We can view them using MView, and we can do tcoffee. So from EBI, you can actually do a lot of the ones we talked about: the tcoffee, the Muscle, the MAFT, and the cluster omega. I do think they used to have cluster W, but cluster W has been retired, I believe. So don't think you can do that with the graphical interface anymore. You can do it with a command line local tool, but you probably are not going to want to do cluster. So what we can do here is we can either use example sequences, which we're probably going to do, or we can use our own, paste in several sequences together. So actually, let's do this. I'm actually going to use the sequences I put down before, because I want to see how these are handled. So we take the distal tail fiber for T4, copy that. We can go to the next one, EP01, distal tail fiber, copy. And then we can add our last one, which is the C6, since we can do 3 sequences now instead of just 2. And as a general note, we're just doing this in a FASTA format where you use a little arrow indentation here to start your sequence, add your sequence name, and then your actual sequence. Here, there's not a ton of options you can change, but you can decide how you want the output to look. I like the cluster W output. You can also change how you want this. You can tell it what kind of output tree you want. I'm going to do second iteration, just so it gives the second iteration. As I said, Muscle uses like multiple iterations of going through and building guide trees. It shouldn't take long. Yep, there we go. So we have, again, something that looks like what we saw with the Needham and Wunsch algorithm when we did on EBI. We have our alignment, and the stars indicate, the asterisks indicate 100% identity. So these are regions that have good identity. The double dots represent regions of high similarity, despite the fact they're changed, they do have high similarity. And then the gaps indicate change, and the single dots indicate more divergent similarity. So this is just the basic alignment, and we don't have the numbers on this output, but you can see we have all three. We can go to results summary that gives us more information, or we can download. We can download the input sequences. This is what we put in. We can download the output. This is just the computational language it uses. We can download the alignment and cluster format. Since cluster was so popular, this alignment format CLW can be used with a lot of different programs. So you can download this and upload it in another program if you prefer. You can look at the guide tree. This again is something you can open with, save this as a text file and open it with a tree viewer. As a phylogenetic tree that it built, again, using that same language. And you can look at the percentage identity, the identity matrix for these proteins. So like the matrices we've been using before, this just shows how closely they are related in terms of identity. So tail fiber T4 is 74% similar to C6 and 78% identical. I'm sorry, not similar. 74% identical to C6 and 78% identical to EP01. You can look at the guide tree that was made. So this is just going to give you a quick overview of how closely muscle thinks your sequences are related, and you can get some information about your sequences this way. Again, you can transform it to whatever you feel like. I was going to do a phylogenetic tree, which is, the guide tree is not a true phylogenetic tree. It's based on the distance matrices, but this one is built using a neighbor joining method, which is more accurate for phylogenetics. Then we can look at result viewers. This is just a way to basically take your sequence or your alignment that you just had and move it into a different program. JalView hasn't been working for me for a while, but there is a standalone version of JalView that's really good for looking at alignments. If you want to go to jalview.org and download it, oh, I see, those are no longer, yeah, okay, so you have to actually download it from the website to view it, but you can export this and view it in a standalone program. Same with MView. MView is a program that the European Bioinformatics Institute has, excuse me, that is good for viewing multiple sequence alignments. And then you can build or look at the phylogenetic tree in simple phylogeny. And you can look at submission details. So all this information you might want to keep if you have, I generally just save the web page completely along with all the information. If I run it through a web page like this, just so I know what happened or know what I did, if I get a different alignment somewhere else, or if I need to go back and check how things were done. But this is a really good place this is a really good, simple place to do a multiple sequence alignment. So if you don't know where to start, you don't know which algorithm to use, I generally recommend Muscle for people. And if you want to use if you need to do a larger alignment, I recommend MAFT. So let's get back to that. Yeah, so if you're just starting with alignments, I recommend Muscle since it's so fast and it is very accurate. And if you need to do a very large alignment, hundreds or even thousands of proteins, some of the faster progressive MAFT algorithms are really good for that. Okay, we are like 75% done. Now we get to do probably the most complicated and hardest to wrap your head around, the hidden Markov models. These are the newer of the algorithms, but they are probably the most accurate and they're really good at finding distant homology. Basically, a hidden Markov model is just a general model for modeling. It's a general way to model probability. So hidden Markov models are not just used in biology. They're used in a lot of different sciences and mathematics. When we're talking about sequence analysis, one thing we can do is look at it as if we were putting together a multiple sequence alignment and then looking at the probability at each position that we have a certain residue. So this is just a simple 5 amino acid alignment of random sequences I made up. So what we can do at each position, skipping this insertion, we'll come to that later, where we have majority field, we see that we can assign a probability for each of these residues at this position. So for example, here, you have a 100% chance of getting P based on this alignment. Here you have a 100% chance of getting K, and you have a 100% getting K when you do get an amino acid, and a 25% chance of getting a deletion. Where here we have 4 different amino acids, so you have 25% chance of getting each of these. Here you have an insertion, so we'll talk about that in just a second. And then the last site you have 100% chance of getting YIM. So what this really is think about it as taking like a journey using a map. So your start position, start of your protein, and the ending position. So you go here, and then here, and then here, and then here. You just move along this route. But depending on the protein, you can do, depending on the protein, you can have different paths you take. So in like this case, there is a deletion here. So when you're moving along this path, you have a 75% chance, according to this, of going to here, to a K, and a 25% chance of coming to a deletion here. And then again, according to just this alignment, you have a 100% chance of going back up to one of these 4 amino acids. And the same with an insertion. When you're coming along here, you have a 75% chance of going to here and a 25% chance of going to an insertion and then going back here. And now this is just based on what we see here, but we can actually expand this and make it more general by adding these deletions and these insertions at each point. So for example, we can go here and we can add a deletion node here and an insertion node before the first amino acid, because there's always a chance you're going to not go to the first amino acid, you're going to go to a deletion. There's always a chance this can be deleted. There's always a chance you can go into an insertion. There's always a chance you can go to an insertion and then the deletion. And there's a chance you can get multiple insertions, just a recurrent insertion, even if that probability is low. So this is just building a model for that. And so you can fill this in for every single position and in between positions to model the probability of basically anything possibly happening here. So this is why I said I kind of think about these as like maps when you're taking a journey. Say you're going down I-40 from Los Angeles to Wilmington, North Carolina. You can start here at Los Angeles and going to Wilmington. That's the entire breadth of the United States. What you're looking at is which proteins or which people, if we're talking about people in maps, took the same route. Because there's always a chance you can just stay on I-40 the entire way. Or maybe you get off and you take a detour, you go visit something, and then you get back on I-40. Like, would you call those the same route? And that's how you identify which proteins using hidden Markov models have followed the same route when you're comparing just 2 proteins. And when you're comparing a multiple sequence alignment, you take all of this information together and you create what's called a hidden Markov profile. This profile incorporates all the information from this multiple sequence alignment and all the probabilities of the different things that could happen. Okay, so these are really complicated and this can be represented mathematically, but for our purposes, this is really just, I think, all you really need to know about this. It's a general way to map the probability of how each protein takes a route from the start to the end. And like I said, you can use this to do a pairwise alignment. And in that case, you're looking at which path each protein took to get from the start to the finish. And are those significantly similar that you can call them the same route, the same protein, related proteins. Whereas if you take a multiple sequence alignment and take all the information you get out of that, you can build what's called a hidden Markov profile. Now, the original paper for this using multiple alignments was back in '95. So as you can see, it's the newest of the papers. One suite I like to use, there are several different groups of hidden Markov model tools, but one of the best and most basic for web use is the European Bioinformatics Institute's Hammer suite. So they have 4 tools there. They have pHammer, HMM scan, HMM search, and jackhammer. By the way, if you're looking at programs and it has hammer somewhere in the name, it probably uses hidden Markov models. When people make these tools, they love to add the name hammer to it. And all these tools use different inquiries and searches against different databases. So if we're going back here, once you build an HMM profile, what you can do with a new sequence of query, you can search that query against these profiles and do some math and statistics to figure out whether or not that protein fits within this profile close enough to be considered part of this profile, part of a related protein here. And this is why it's good for finding distant homology, because you can have like deletions in the middle, but it can still find like the important parts that are found in throughout most of the protein. So for example, if you had a multiple sequence alignments where you 100% identity here, 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 and here, but you had some gaps in between, the other programs we talked about may struggle to find that homology because there's so many differences. But hidden Markov model is really good at finding those hidden connections because it's based more on like probability than just straight up what aligns, if that makes sense. So with the tools from the European Bioinformatics Institute, we have the first one that you can use to search a protein sequence against the protein database. And that kind of does what I talked about, how you look for proteins that follow the same route. We also have HHM scan, which searches protein sequence against a profile library. So this is a this is a library of HMM profiles that have been predetermined or pre-calculated based on multiple sequence alignments. You could also use HMM search to search for protein alignments or HMM profiles against a protein database. Now, this one is really good when you're looking for proteins that may fit a group of proteins you already have. And then jackhammer, I think it's called jackhammer because jack of all trades, jackhammer. This you can search a protein sequence, a protein alignment, or an HHM profile against a protein database. And I've got another table here showing you like when to use each and what they're good for. It said the first one, the p-hammer, it's good. It searches a single protein against a protein database. It's fast. It is not as accurate as the other types, but it is fast and it is generally reliable and accurate. That HMM scan is good for looking at protein sequences against protein domain profiles. So if you look at a lot of like PFAM, for example, the PFAM database, they have collections of protein families, and those protein families are based on calculations of HMM profiles. And this is what I meant earlier when I said I want to show you just this handful of algorithms, because you can then take what you know about these algorithms and use that knowledge to learn about all these other tools that you didn't really even know were related. So a lot of the protein family databases use HHM profiles to build these databases and search your protein against those databases using hidden Markov model algorithms. Okay, so yeah, so this HHM scan is good for identifying proteins with known protein domains against a query. So basically what you're doing is you're taking hidden Markov model profiles that have been generated for domains or sets of domains and proteins that have categorized them into families. And now you're searching your protein against that. And so this is a good way to find out what domains are in your protein. Now, HHM search is actually really cool too, because what this does is it searches protein databases against your profile, HHM, and you can generate that with HMM search, ham search, let's go ham search. You can do that by actually putting in a multiple sequence alignment. You don't actually have to, or multiple proteins, you don't actually have to have a profile made previously. So what that means is that if you have a set of proteins that you're interested in, say you have like phage tail fibers, and you know they're all closely related, you can align them all with multiple sequence alignment algorithm, or you can just put them into HMM search, and then you can search a database of proteins against that set that you put in. And so what that does is identify more proteins that may be related to your group. And so you can expand that group that you're studying. So that's a really powerful tool if you're looking for like, proteins with similar, or phages with similar tail profiles. And it is more sensitive because profile HHMs are really sensitive compared to just like dynamic program or blasting. So that's one reason we like to use the profiles. And then the jackhammer is very slow compared to these others, but it is really good at finding hidden things and distant relationships. You can take a protein sequence or multiple protein sequences and build a profile, or it can take a profile, as I said here, can use any of these, and it will search against the protein database. So what it does though is when it takes your sequence, and then it takes the hits to that sequence, and then it builds into a profile from the hits. Then it searches the profile it made against more. So what will happen is you'll add say if you add a protein sequence to search against other protein sequences, when you get hits, it'll align them and build this profile. Then it will go and take that profile and search the database again against that profile to find hits that are not readily identified from a single protein, but may statistically fit in with a group of related proteins. So in that way, you can find things that are not readily obvious to build a to identify distant relationships between a group of proteins. So say you have a phage tail fiber again, and you want to find proteins that are related to it. You can use Jackhammer to first do a protein search, a protein query search against a protein query, and then you can I'm not sure about that. Then you can build a HMM profile from that as it gets hits. So if you take a T4, it's kind of slow, so we can't show you here, but say we took our T4 tail fiber that we've been working with so far, we search it in Jackhammer against the protein database. It takes those hits, it aligns them with the query I put in, and it builds a profile. Then it takes that profile and it searches against that database. And then it builds a new profile with the new hits, and then it searches the database again. And it will do this iteratively until you either tell it to stop or it reaches a convergence. So these are really powerful tools for finding some distantly related proteins that may be related to what you have. We can look at these tools, which we are, because we still have about 50 minutes left. Let me get this off. If you want to check it out too, you can use, again, the QR code, or again, the European Bioinformatics Institute under Tools, the hammer. In the slideshow, and go there. So when you bring this up from Tools /HMMR, you can do the quick search, which I believe is just going to be the P-hammer search. But you can also go to click the search tab, and this gives you the 3 I talked about, or the 4 I talked about. So you can start with P-hammer, and we can add an example. You can just put in a single protein like that. And what we're going to try to do is we're going to see if we can actually search our protein, the one we've been looking at. We're going to submit this, and then we're just going to open this again while that loads. Open this again while that loads, because this site is being updated and they're doing some service on it, so it's been kind of slow, and sometimes it doesn't like to load correctly. So while that loads, I will walk you through the other ones. There are some other things you can change in terms of options. You can, like with BLAST, you can restrict taxonomy. So, if you wanted to restrict it to like Escherichia, you can do that. And now this is going to set it to search for that only. And you can also do predefined representatives. So if you want to search viruses, you can do that. It shows you what viruses it has. You want to search for bacteria, you can do this. You can choose which bacteria you want to search for. So you can limit your searches and increase your speed. You can also choose which database you want to search for. Again, this is going to be something you're going to have to kind of look at on your own, decide what you need. These are all really good databases, but some of them are better than others. I do like that they have added the AlphaFold database, because that means all your hits are going to have a structure that it might, or it's going to have a structure that you can view for any of your hits. But a lot of the reference proteomes also have that, because the AlphaFold database has a ton of protein structures. We're going to see the same options for most of these. The only thing I also want to point out is the E-value, where we talked about expected value. I introduced that with BLAST because it's used in a lot of programs as well. The expected value, again, is just chances that this happened at random. And then the bit score, again, is just your alignment score, your normalized log alignment score. So let's see if this is, yep. So remember, we searched with our T4 protein. And up here, we see domain matches from PFAM. So we have a domain hit for phage tail fiber repeat, the phage tail fiber collar domain, the tail fiber repeats, and we also have disordered. It also predicts disordered when we search for it. So this is just something that it adds. Blast also does something like this. And then we can look at the hits. So luckily, the best hit we got was from T4 phage, which is the protein we're using. We hit this arrow here to expand and look at some of the statistics, including identity, similarity, bit score, and e-value. We can look at more distantly related ones as we go down. We can also choose which columns we want to see by hitting customize. And so you can choose, we want to see how identical they are. And we want to know whether or not there's a known structure. So we can add that, and that just adds the known structure. So if we want, we can click on this, and it will take us to the protein database archives for the structure for the T4 long fiber. And so we can look at that if we need to. We also have a lot of cross references here. You can look at the coding regions for your hit and the sequence. You can look at the Interpro and PFAM databases. These are protein databases that you can get. You can look and see what family your protein belongs to, and then look at other members of that family. And yeah, that could be useful for a lot of people. You can look at interactions. Actually, I'm not super familiar with Interact. You do have a lot of additional cross references here with EBI that you can use, which is really helpful, especially if you're trying to search for more information for your protein. I wish I was aware of some of these tools and cross references when I first started. Now, this is where it's going to get a little hairy because taxonomy doesn't always like to load on this, especially recently. Here, well, maybe it's going to load. Yeah, it does. So here you can see the distribution, the taxonomic distribution of your hits. So almost all of our hits were in either bacteria or viruses. And we can click on these to expand them and get a better idea of where they are. And see Myroviridae, yep, that's where we would expect them. And then we can just keep going until we find out what we want to, find out which hits we have in terms of taxonomy. You can go down here since we expanded it, and it will show the hits and the organism that you're getting the hit for. We can also look at the domain tab. Let's hope this loads. We're not going to look at Interact because I actually am not super familiar with that. I've never used it before. Okay, yeah, this did load. So this is a really interesting feature with EBI. It shows you the hits that have certain architectures. So this says that we have 111 sequences from our hits that have this domain architecture, which means they contain this phage tail collar domain. We have 54 hits with no known architecture. Then we have some here with a chaperone. This is interesting, chaperone of this enzyme, which this is a type of enzyme, endosalidase, that cuts types of sugars, I believe. That's not unusual to find not sugars, proteins on the surface of bacteria. So this is something interesting you can look at. If you wanted to, for example, show all, you want to see which one have these peptidases, which is not uncommon to see in phage tail fiber, right? Because they can break down the cell wall or the peptidoglycan of the surface, on the surface of the bacteria. Sugars. So yeah, this gives you the different ones that different hits that you can go and search those. So yeah, there's a lot of useful information, a lot of useful tools you can find from these types of alignments to expand what you know about your protein and what you know about related proteins. And of course, you can download your the information in different formats. Okay, so we looked at hammer. We have 40 minutes left. So we're probably not going to go over these different examples, other than we talked about HMM scan and how you can search protein sequencing against profiles. That is that's useful for like domain searches, like I said. And then the HMM search, this is good for searching for alignments or profiles against protein sequence database. So if you have several proteins and you want to find more proteins that are related to it, you can search with something like this, either an HHM scan or jackhammer, and this will find members of basically the group of proteins you have. So if we wanted to use like, these distal tail fibers, we could search for all of them, and it's going to be more powerful in finding hits than just searching for one alone, because it's searching for the pattern, these HHM profiles we talked about. So, okay, we can go back. So this is a very, very useful tool. There are several HHM suites. Another one we're not going to talk about, but is really powerful, is the Max Planck Institute. That is really good for searching for, again, really distant homology. They're HHPRED and HHBlitz programs. Now that we've covered the basic algorithms and we have enough time left, I want to actually introduce a few more of the more advanced BLAST programs. We're not going to be as exhaustive with these as we were before with like the dynamic programming, because it's built upon the same ideas that we've already talked about. And I hope you're starting to see how the basics we learned about so far are starting to build into more advanced concepts. So the first more advanced BLAST is the PSI-BLAST, the position-specific BLAST. This is good like HHMs, hidden Markov models, for finding distant homologs, and it is iterative like a lot of the HHM programs we talked about, like the HHMs or the jackhammer. Now, this works by first running a BLAST-P against your protein, creating a multiple sequence alignment of hits. Now we know what a multiple sequence alignment. And from this multiple sequence alignment, it generates something called a position-specific scoring matrix from this alignment of hits. Then it uses dynamic programming to search this PSSM, this position-specific scoring matrix, against the database. This actually works a lot like jackhammer in that way. Remember, jackhammer, it takes your protein, it searches against the database, and once it finds hits, it creates a profile, then it uses that profile to search for more hits. Well, PSI-BLAST does something similar, but instead of using hidden Markov models, it uses position-specific scoring matrix, which we're about to talk about. And it does the same thing. You put your sequence in, it searches against the database. When it finds hits, it creates this profile with position-specific matrices, and then it searches for more sequences that fit into that profile. So a position-specific scoring matrix is a lot like the substitution matrices we saw with the PAM and the blossoms. So recall that this is just a log transform matrix of probability of going from this amino acid, like on the column, to the amino acid in the rows. It's just a way to model probability and to look at whether or not something's neutral or positively selected for or negatively, so that you can get an accurate substitution matrix when you're building alignments with dynamic programming. A position-specific scoring matrix actually works a lot the same way, a lot like this, except instead of having amino acids against amino acids, you have amino acids against positions. So what happens, how you build this, is by getting a bunch of hits and creating a multiple sequence alignment. Now we know how to do that, the progressive and the iterative alignments. And once you build this, you can then create a position-specific scoring matrix that shows what is the probability of having each different amino acid at each position in the alignment. So here we have 10 positions and we have 10 columns, because each column represents a position. So for example, like the second position, we have lysine. We have mostly lysine, so we have a high probability of a lysine here. We have a valine, so we have more than zero probability of a valine. And doing this, this is how PSI-BLAST is able to create a position-specific scoring matrix. So it runs a regular P-BLAST that we talked about. It takes the hits, it lines these up, and then it creates this probability matrix based on what it sees in the alignment. Then it uses this alignment, or this profile, because this is a profile just like the HHM profiles. This incorporates all the information from the multiple sequence alignment. It then searches for other things in the database that match this profile. So in that way, you can find distant homologies without looking for like specific matches in the alignment. And like jackhammer, it's iterative, so it builds upon itself. Once it has this PSSM built, it looks for proteins that statistically match this same profile. And if they match, it recreates a multiple sequence alignment, creates a new position scoring matrix, and looks for more hits. And it keeps doing that until it either has a predetermined stop point or it reaches stops increasing the amount of proteins hits that it gets. So how does this work in practice when you're doing, again, our favorite little matrices by hand? Basically, I've only cut this off because we have 5 positions, but instead of doing, when you're aligning a protein, instead of aligning a protein against a protein, a peptide against a peptide, what you're doing is you're scoring a protein based on what it has in each position. So in this example, what you would do is you would do the same thing as you're filling out your dynamic profile, and you look to see what the score is at each position. So for example, we have an E here. So we look over here and we find blue. So we have 5. So when we're scoring this cell, we use our trusty scoring scheme and our 3 calculations. We move over 1, up 1, 0, plus the match or mismatch from the PSM. In this case, we have 5. Put a 5 there. Put a 5 there. And we put an arrow where it goes, because going here would be -4, going here would be -4 because of the gap penalties. Again, it's the same equation over and over again. And so we can fill out the entire matrices, the matrix, this way and build an alignment to see how well this sequence fits our profile, our first 5 positions in our profile here. And so in that way, it's really good at finding distant homologs to your protein. So what you can do is go into SciBlast. You can search for, say if we want to use the T4 again, you can search for that. It'll find matches for it, and it will give you a chance to select the ones you want, or you can tell it to choose certain scores, and then it will rerun it again, where it will run the PSSM against the database, and it will find additional hits that could match that PSSM profile. So hope that's clear. Now, the next type of advanced blast is called the RPS blast, the so-called reverse SciBlast. This one, a lot like some of the HHM searches, is good at finding conserved domains. Now, what this does is align your query sequence against pre-generated PSSMs. And this is how the NCBI's conserved domain database works. So the curved domain database uses RPS blast to search your protein against these position-specific scoring matrices that have been developed against different domains and different conserved domains and proteins, to see which of those profiles your protein matches with. Yeah, so the NCBI's conserved domain database is really good for this. R uses this. It takes database domains curated from a variety of sources, including SMART, PFAM, COGS, TigerFAMs, PRK, and we have time to go over this one really quickly. But one thing I do want to note is that all these databases are built using the algorithms we've already talked about, either hidden Markov models, which I hope you understand now, or specialized blast, which I hope you understand how they work now. So this is what I meant this is what I meant when I said I wanted to start with just a handful of algorithms, and hope that understanding those will help you in the future in figuring out new tools or ones we haven't mentioned today, because they all kind of converge around similar themes. Okay, so I'm going to try this. We're going to do this one really quickly. Let's see. I'm going to go to this website. This is the conserved domain database. We can just paste in our T4 distal tail fiber. You can also put a nucleotide or a session or gene ID or any sequence really. You can also do, there's also a multiple protein. There's also a batch version. This allows you to put in multiple proteins at once, and it doesn't search the way, it doesn't search against a multiple sequence alignment. It searches multiple times against your different proteins. So you put all your proteins in and search them together. But there are some, there's a few options here you really don't need to mess with. You can like choose either the conserved domain database, which incorporates all of these, or you can choose which one you actually want to search from those protein databases we talked about. So you can see how many position-specific scoring matrices each of them have, but search for all of them using the CDD, which has the most because it incorporates all of these together. You can set the expected value threshold. So if you don't want any results above a certain level, you can cut those off, and you can apply low complexity filters, but you don't really have to do that because of the statistics adjustments they have added. And you can force a live search. This just means that if it detects that this search has been performed before, it'll give you those results rather than searching again. Yeah, you can do a few other things, but really you can keep all of this the same. You can just hit submit, which you add your protein sequence, and you should start to get something that we saw before. Because actually this is searched when you do a blast, a protein blast, you'll get something that looks like this. Recall how we had, actually this identical graphic at the top of a protein blast earlier in this workshop. And from there, we can get more information by just hovering over to figure out what this collar protein does. Regionally found in conjunction with certain PFAM families, blah, 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 blah. Get more information about this. And get the E value. It's a pretty high E value, but it still works. Shows you the interval that is found in your query. You can expand this, which is just the same information that's up here. It'll show you the sequence that is on in your protein and the sequence that it is aligning to in the database of domains. So you can see exactly how your sequence fits in what it's saying it's related to. And you can search for similar domain architectures by doing this, by just clicking this. Basically, it'll look for proteins that have similar architecture, which is better when you have multiple domains than just this one. And you can also expand this so that you basically include more information in the results. But honestly, all you really need is just this information here: your bit score, your E value, and the domains you search for. So this is a good way to find what domains are in your protein. And like I said, when you're doing a blast search, or a P-blast search, this only takes a second. You'll see what I mean. This will come up at the top of your search results. Should I take a few seconds? But yeah, this is not so good for when you already know what your protein does and you know what the domains that are in it. But if you have a new protein and you're trying to figure out more about that protein without doing a bunch of experimental work, like I said before, it's all about relating what we don't know to what we do know. Then this is a good tool to find quick domain searches. Let's see. So I think that's all I want to show you with this one, but I do want to bring up another tool I like using, which is from Japan, the Kyoto University Bioinformatics Center, which is called Motif Search. Let's do this here. This works in a similar fashion to the conserved domain search. Yeah, see, when we search for the T4 in BLAST, we get the same graphic because we're seeing the same search against the domain conserved domain database. Now this motif search is a little simpler. You can just add your sequence here and it does search against the database you tell it to, either the NCBI's conserved domain database. I think this is an older version that doesn't include PFAM. You want to do PFAM separately, and you can include prosite patterns and prosite profiles. So you just compute, do a quick search. We're leaving everything else the same. And this should only take a few seconds. And this will search your protein against these databases to search for known domains. So you get your query here, along with where your hits come from. So we have hits. We have only one hit from NCBI. We have multiple hits from PFAM, including these hits. And we can look here to see where the tool came from. NCBI gave us one hit, which we saw before, the phage collar tail protein. Can hit show more, show all, to get that the same information we saw here. And it gives you quick details about the E value and the bit score based on what it has here. We also have the information from the PFAMs. We don't have as much information, but we can go and click on these and get more information. Go back to filling domain. We click on this will take us to the domain entry for this protein domain. And we can get more information about it and figure out which sequences contain this and more information about what it does. So again, really good tool for finding information about your protein. And you can also hit details. I'll show you again. Hit details. It will show you your query along with the highlighted region that matches and an alignment of where it matches to what the database hit was. And yeah, I think the last thing I want to mention is that it's really good that it shows the A value, because again, this is like what the chances that happen by chance. And this is a good example because if you look at the phage tail fiber here, even though we know this is a phage tail fiber, these repeat hits are probably low complexity. And so we hit them several times with a very high E value. I mean, this one is approaching 1, so we would not call this significant. And this is probably because they happen often. And same with the back to film, actually. It's a very high E value. The collar protein, though, we have a significant hit with, just as we do here. Okay, I think we are getting to the end of what we want to talk about. Yeah, so this is how the conserved domain searches work. They use a reverse SciBlast, which uses the position-specific sorting matrix. So that's how the NCBI conserved domain works. So you can use that in conjunction with the HMM profile search to get more information about your protein. Sometimes it will show, one might show something, one might have hits that the other one doesn't because they're different methods. It's always best to use more than one method whenever you can. We're getting to the end of the specialty blast. There's another one called PhyBlast. This is a pattern hit initiated blast, and this is good for finding functional homologs. So instead of searching for just pieces of your query, it searches for user-defined patterns. So what that means is that we'll just go to real quick, we got time. When you go to NCBI blast, you go back to here and go to protein blast. These are the specialty ones we talked about. A PhyBlast is where you still put in your query, but it searches for a pattern. And this pattern is just going to be some kind of syntax using it's going to be a protein pattern. And so you can go on here and click on this, and it will, I thought it was there's some information on Blast about the syntax to use. We're not going to go over this exactly, but if you know that you have a pattern in your bacteria, that maybe it's not necessarily a domain, but it's found in all the protein, I'm sorry, not bacteria, protein that you have, if you have a pattern in your protein, and you want to find other proteins with that pattern, you can enter your pattern here and it will give you hits adjusted for hits that contain that protein sequence that you have. And because it has a little more advanced syntax, you can add things like gaps or, any amino acid. So I believe it's like X is for the any amino acid and maybe the star for gap. I can't remember the syntax exactly, but you actually create this pattern that you put in here and it will search for that pattern in the hits. Excuse me. So that's useful if you have a pattern you want to search for. You want to find more of those patterns. Excuse me. Okay. So where are we now? PhyBlast. Now the last one is Delta Blast. The domain enhanced lookup time accelerated blast. This is good for finding structural homologs. And it aligns your query to the conserved domain database, and then it calculates PSSM from that. So it works a little different from the RPS blast in that it builds a profile after your protein gets a hit in those conserved domain databases. So again, it's kind of like Jackhammer in that it can build a profile and help you find other proteins that may fit within that profile. I got another one of my tables here to give you kind of the primary uses and the descriptions and when to use each of these different blast types. I don't feel like I really need to read this slide. I think we've gone over everything. Feel free to take a picture of any of my slides. I guess it's a little late to tell you that, but yeah, feel free to take any pictures. This is also being recorded, so we are going to put this up, I guess, on the website. I'm not really sure where they're going to put this up, but yeah, you can go back over and get any of these tables you need. Okay. I also have a software list of things we talked about, and hopefully you have at least somewhat understanding now of how all these different tools work. They all have their own modifications and their own quirks and their own things you can modify, but I hope that you now at least understand how a lot of these work. We went over all of these in this workshop, from the dot matrices, and I don't know why I have an S in matrix. That should be matrices. Dynamic programming locals, progressive MSAs, the iterative multiple sequence alignments, and then the different types of blasts, the nucleotide, the protein blast, the nucleotide protein mix blast, and then the hidden Markov models. With these softwares, you should be able to get most information you could want from your protein. You should be able to build databases of similar proteins. You should be able to do a lot by using these tools we talked about today. Now we have a little extra time, about 20 minutes. There's another tool I like to tell people about that I really, really love. It's called Paper Blast. Now, again, this goes back to how I hope you can use the basics we talked about to learn more about the more advanced stuff. Paper Blast uses BLAST-P, so now you know how that works, to search against the database that they've curated from open source papers. So basically what they've done is this group has taken proteins that are found in open source or open access papers, journal articles, and they've created a database with those. So what that means is that you can go here and go to their website. Oh, that's the wrong one. It's the next one. That's the paper. You can go to their website here, and you can enter your protein sequence. We're going to use again our T4 distal tail fiber. Enter the protein sequence, copy it, paste it, and search. It's really fast. And what it's going to do is it's going to search all these open access papers for proteins similar to the protein you gave it. And I absolutely love this tool when you find a new protein or a new protein of interest, or a new gene of interest. What it does is it shows you the hit for the like the database codes for these hit, and we have a long tail fiber, GP set 37. This is exactly what we put in. And it tells us here the percent identity and percent coverage in basic terms. You can click on that to see the alignment, and with your identities and your positives, and you can see the entire line by just clicking on it. But it didn't show you the papers that this protein is found in. Not only that, it shows you a little snippet where they talk about that protein or where they mention that protein based on the accession numbers. And it also looks at ones related to it, like this long tail fiber is from another enterobacterial phage called K3. We can again look at the coverage because we've done a protein blast. Look at the positives and the gaps, and we have 70% identity. And we can look at papers for that. And now if we want to actually look at this paper, we can just click on that, and it takes us to the paper so we can read more about it. So like I said, I absolutely love this tool when I'm getting new, when I've come across a new protein. It's one of the first things I check just to see if there's any information about easily out there about my protein that I just found, and how I can relate to proteins that we already know something about. It's already been published on. And you can keep going. Excuse me. You can keep going all the way. There's several hits all the way down to, you're like 20% coverage. You can, if you know more about your domain architecture, you can find out where this is at in your protein and see where it relates. For example, we're looking at a distal tail fiber. So this means that you could be looking, if we knew where it actually bound to the bacteria, we could look for which of these hits happened at that end and find phages with similar tail fibers with maybe similar host receptor domains. So like I said, I really love this tool. It has other things you can do with it. You can look at functional residues by going to Site Blast. And again, now you know how BLAST works. So I hope you understand, at least intuitively, how some of these tools may be working. And so this looks for sites where things bind, such as iron ions and carbonate ions. I believe I'm not super familiar with this tool, but I believe this is based on structural analyses and structural biology. You can again look for conserved domains. This, I'm not sure. Yeah, this relates you back to the conserved database, which we already talked about. You can find matches in UniProt, which is another database of proteins. You can compare it to structures. You can do all this kind of stuff. It's really cool. So I would recommend using this paper or this tool if you're working on a new protein or when you come across a new protein. I do love it. Okay. Another tool that's really good, especially if you don't have access to a lot of these tools locally, or you don't want to use the web for some reason, are all a whole bunch of different tools. And sadly, I didn't want this to happen, but my workshop had to coincide with Jason Gill's workshop on the Center for Phage Technology's Galaxy server. Center for Phage Technology, they're an old hand in phage research, a legendary group out there at Texas A&M. They have a really, really nice Galaxy server. And Galaxy, we're not going to go over this. There's tons of tutorials out there, and they have tons of tutorials. But they have this nice server where you can actually use a lot of the tools we talked about today. So they have all the tools they have over here are in different subcategories, and you can search. For example, if you want to use math, they have a search here for that. Math. So if you can log in and you have your own database of things you'll keep, you can then add your data to this and run it on their servers. You can choose your matrix selection that we already talked about. You choose your coefficient for your Blossom matrix, where we already talked about Blossom 62. All these things are starting to become more and more familiar. And so you can run a lot of this stuff on their server, and you can create workflows where you can string several tools together. I'm not going to go over that because we don't have time, and I'm not an expert with Galaxy, but they do have some really good tutorials too, and they have a lot of good workshops and videos you can check out. So I do encourage you to look at their servers if you get a chance. And let's see. And yeah, here is my my software list. I went through and tried to find out which software were found on their servers, and almost all of them are. A few that aren't, but most everything you want to do is found on their Galaxy server. And I think that's it. I think we made it. It's 14 minutes to spare. Thank you guys for taking the time to join the Zoom. I am very sorry I couldn't be there in person. I really, really wish I could have been. I had an amazing time last time. It's such a great place, and Benny's amazing. I got to thank my lab, the Moresso group, and Taylor. And I've highlighted the ones from the group who are there that weekend. So please do try to talk to them and hang out a little bit. Yeah, and I also, again, I want to thank the organizers of this conference, and Ria, Jan, and Jessica. Thank you so much for working with me on this, and I do appreciate it. So if anyone has any questions, I am free. Feel free to email me. Feel free to drop me a line. I'm seeing we getting some text. Let's see what we got. Thank you. I hope it was informative. And like I said, they're going to be sharing this if you need to go back and look at anything or take some pictures of the slides. I may talk to them about putting some of these slides up on some kind of shared PDF or something. But yeah, I guess that's it. Thanks, guys. If no one has any questions. Enjoy the rest of your week.